then let's start chapter 4. Okay, so chapter 3, we introduced you to, maybe for the second or the third or maybe fourth time, we introduced you to Mendelian genetics. In chapter 4, what we're going to do is we're going to continue talking about Mendelian genetics, but we're going to do it in a way where we relax some of the simple setup of the original Mendelian model, right? So, who can remind me what the original Mendelian model was? We had two of them, right? We had two different models we looked at. There were two different ratios that pertain to each of those two models. What was the simplest Mendelian/genetic model we've dealt with in this class? What is it? Monohybrid. And so in the monohybrid, what were the assumptions? Good, Maya, what were the assumptions of the monohybrid cross? Or when, what was it? Well, first of all, what was the ratio?
3-to-1 ratio.
Okay, 3-to-1 ratio, good. When we see that 3-to-1 ratio of dominant phenotype versus recessive phenotype, what does that tell us about the system? What do we automatically kind of know? I mean, because that's the whole key here, right? We're using these classical genetics, transmission genetics, looking at a ratio and then inferring things from that ratio. Corey, what did you say?
It's independent assortment.
Okay, so, well, hold on to that one for just a second. 3-to-1, how many genes are we looking at? One gene. How many alleles? Okay, so you guys know this. What's the interaction between the alleles? Dominant recessive. Okay, what are the alleles doing? They are, what are they doing independently? Separating, or the other S word? Segregating. Okay, so that's the first one. The second one has to do with Corey, what Corey said. What? Independent assortment, the principle of independent assortment. To test that principle of independent assortment, we look for a ratio of what? 9-3-3-1, good. When we see that ratio of 9-3-3-1, what does that tell us? How many genes are we dealing with? 2. How many alleles at each gene? What's the interaction? 2. What's the interaction of those alleles at each of those genes? Dominant recessive, right? And when we compare the two genes, we see that the alleles at those genes are sorting independently. Okay, so those are the two really simple models, right? Now, for this chapter, we're going to extend that. And we're going to start to say, okay, what happens when we don't have this simple dominant recessive interaction? What happens when we have more than one allele at a gene? What happens when we have more than one gene and the genes are affecting each other? So that's what this whole chapter is about. It's just making it a little more complicated, right? But that's kind of how we do it in science. We start with a really simple system. We look at that system. We try to gain some basic knowledge from that system. And then we take a step beyond that to make it a little more complicated and try to understand more and more complicated scenarios. Okay, so here's an overview of what we do. We're going to look at allelic variation, gene function. While we're relaxing those assumptions, we're then going to look at gene action. From up until this point, one of the simplifying assumptions is that we just simply say, okay, genotype is a single genotype directly corresponds to a single phenotype, right? We're going to look at when that isn't always true, and we see that that isn't always true in pretty much all of the cases, right? And then the last part, we're going to, again in this chapter, go back to human genetics, and we're going to look at pedigrees, and we're going to calculate inbreeding coefficients and coefficients of relatedness or relationship, and do a few more things with the pedigrees. Okay, so here we've already gone through the assumptions of the Mendelian model. I thought that was the next slide, so never mind. It's the next slide. So here, this is a bell curve, right? What kind of distribution? For people with staffs? Yeah, normal distribution. We're showing it up here. Let's see, what are the data here? All students taking a test, scores in percentile, right? You could also probably use this bell curve if we were to go out and survey or measure all of the students in the College of Science as they were leaving the building today, and we were to measure height. We could do that, and we would chart heights on the y-axis. axis, and if we sampled enough people, it would probably look something like this curve, this normal curve. We see this over and over again when we look at natural populations. This is a function of allelic variation, okay? So why do we get this curve? If we were to separate out males and females, we get the average male height. What would the average male height be? Does anybody know in the U.S. population? Probably 5, maybe, I don't know if it's that high. I know does anybody know the country with the highest average height in the world? It's Denmark. Not Denmark, it's Holland. Holland has, there used to be it has, the average height is 6 feet. And that's pretty high. The world, does anybody know, if you look at it across the entire world, what the average height is? Something like 5'6". So I imagine the U.S. So we'd probably be, well, should we take a survey? How many males in here are taller than 5'9"? How many are shorter? Okay, so that's probably not it. How many are taller than 5'10"? Oh, we have a tall group in here. Shorter than 5'10"? 5'11"? Yeah, that's probably where it is. Oh, so we actually have a tall group in here. But so here, this would be your average size. What this tells you is that you have your most frequent here, and then you have a falling away from those frequencies at both sides. Okay, when researchers went and looked at human phenotypes and started to measure different things, we could see that for the most part, if they sampled enough individuals, you would get this bell curve type shape, right? If you were to do that with Mendel's pea flowers, would you have gotten this nice bell curve? What would you have gotten if you were to measure Mendelian characteristics, say flower color? Would you have a bell curve? What kind of curve would you have? Remember the flower colors, purple versus white? Would it have come out like this? No, what would it have looked like? Of the thousands of if on the x-axis we had some sort of measure of color, it would have looked like this, right? Red and then white. It wouldn't have been a bell curve. It would have been this normal distribution. So why is it when we start looking at human characteristics do we get the vast majority of them coming out in a normal distribution? That's what we're going to look at. And that has to do with the number of alleles and the number of genes that are influencing a trait. When it gets more complicated, it begins to take this normal distribution. Okay, that's what I wanted to show you that. So again, what we've done with the Mendelian genetics up to this point is an oversimplification. Here, this is this model I keep making you, or trying to get you all to reproduce. You know, single gene, 2 alleles, etc., etc. You should have that beaten into your heads by test 2. Okay, and I think you will because I'll reiterate it a whole times. Okay, so now let's just go through and sort of in a stepwise fashion, we are going to look at the assumptions of that model. So the first thing that we're going to do is let's look at the interactions between alleles. Let's say, let's ask, are there situations when alleles are not behaving in a simple dominant recessive interaction? And most of you are shaking your head yes, because yeah, we're pretty familiar with some of these instances. The first instance that we're going to talk about is incomplete dominance. Incomplete dominance can be defined by the heterozygous individual. In incomplete dominance, the heterozygous individual has a phenotype that is kind of midway in between one phenotype and the other phenotype. Well, we could say the dominant phenotype versus the recessive phenotype. In this sense, one allele is partially or incompletely dominant over the other. A good example of this is with snapdragons. If you remember when we were talking about, let's go back again and talk about Mendelian genetics or Mendelian phenotypes in the pea plants. Flower color, what were the flower colors again? Purple, remember, purple and white, right? So if you have purple and white, right, those were the only two. In Snapdragon, you actually have red is the sort of the dominant phenotype, white is a recessive phenotype, but the heterozygous is in between red and white, that is pink, right? And so when you see incomplete dominance, a lot of times those genes or the phenotype that results from those genes have to do with an amount of something. So here, with this genotype, what was called big W, it's producing a certain amount. Each of those genes is producing a certain amount of pigment, okay? In little w, those genes are not producing any pigment. In the heterozygous individual, you have one gene that is producing pigment, but the other gene is not producing pigment. So you basically diluted the red into half. And so this is the amount of gene product. This would be 2x, this would be x, this would be 0. Sometimes it doesn't matter. With some genes, it doesn't matter whether very much is produced or a whole bunch is produced. A lot of times those genes are genes that code for things like enzymes. So we know if there's just even a little bit of an enzyme there, because enzymes can sort of recycle their function, we don't need a lot of them. Okay? In this case, maybe it is something that has to do with the actual pigment or something like that. Okay, so when you're looking at phenotypes that depend on amounts, we see that in those cases, a lot of times the heterozygous individuals show incomplete dominance. Another deviation from that dominant recessive condition is codominance. Codominance is different than incomplete dominance because in codominance, you have both of the phenotypes being expressed. It's not a phenotype that's halfway in between the dominant recessive phenotype, but rather it's a phenotype where both of the different alleles are being expressed. And a really good example of this are the different kinds of human blood types. Okay? If you were a certain blood type, and you, let's say somebody, let's not say you, let's say somebody got into an accident and they had a certain kind of blood. Let's say they had type O blood and they were given a transfusion or they were given some blood, right, and it was AB blood. What would happen to that individual? Is anything going to matter? Is it not going to matter? What happens? What happens generally when you are given, or when foreign antigens, or what we call foreign cells, enter your bloodstream? Let's say foreign cells that express certain kind of cell surface proteins. What do we do what does our immune system do? What do we do? Yeah, we make antibodies against it, right? And so let's say here we have a foreign protein that's on the cell surface of this blood that was given to us. Let's say we were desperate, we didn't have time to take, or maybe we were doing this, say we were doing this 300 years ago or 200 years ago or something, before we knew about blood types, or 100 years ago. Well, not 100, but sometime in the distant past, you would make antibodies that correspond to the antigens. What's an antigen? What is it?
Antibody generator.
An antibody generator, where's it located?
Foreign body.
On the foreign body, on the cell surface. Let's call it cell surface protein. So you have a particular protein, your body makes antigens. The antigens, all they do is they bind to the cell surface protein. In essence, what does this do? What's the function of this for your immune system? Why does your immune system want to do this?
Flags them for destruction.
It flags them and then it kind of corrals them all in, right? It flags them for destruction from other cells from the immune system, and then it kind of corrals them all in and gathers them all together. Okay, so that is the function. So let's just say we did it, and this is how we do it. We do it in rabbits. We inject rabbits with human blood, right? The rabbits produce these antibodies. We then take out the plasma from the rabbits, and we now have extracted what we call an antiserum. So we have a liquid that contains a whole bunch of those antibodies. All right. These antibodies correspond to specific cell surface proteins. Okay. So now, what have we done? Why would we want to do that to the poor rabbits? do with that now? One, we could maybe extract the antibodies, right? But what else could we do with it? How many of you have taken physiology lab? Don't you do blood types in physiology lab? How do you do the blood types?
With antibodies.
Antibodies, where do those antibodies come from? Maybe from rabbits, right? And so you have the antiserum. So here you have an antiserum. An antiserum is just this liquid with a whole bunch of antibodies in it, right? You expose that to blood, and by doing that, you can tell what a person's blood type is. Does everybody know their blood type? I didn't know it, and then I told one class, and somebody came in and they poked my finger, and I just forgot. So it's important, but I guess it's not that important, because I guess they test you anyway, right?
Test you.
Yeah, just to make sure. Yeah, I'm not sure what kind I am. How many of you, well, let's show of hands, how many of you know your O type? How many know your A type, B type, AB type? Oh, yeah, Carl's an AB? Okay, so we have a sampling of everybody. And then also, what are you usually? Your O what? Negative, positive. What does that have to do with? RH factor, right, which is an entirely different one. So we have ABO, we have MN type, this is what we're going to look at here. We're going to look at ABO, we're not going to look at RH, but we have a whole bunch of different types of blood. Why is that? Why do we have all these different kinds of blood? What are these kinds of blood really? What, Elizabeth? Yeah, they're alleles to certain genes. Those genes code for simply just different kinds of cell surface proteins. These are just a whole bunch of different systems that are describing the combinations of these cell surface proteins that you have. Okay, so let's say we wanted to test for MN blood type. And so we would produce a bunch of antiserum, right? We would then draw from everybody in the class, right? So we have the antiserum, we draw it from everybody in the class. We then expose the blood to the antiserum, and we look for this thing called agglutination, which is basically just a clumping up of the blood. If the blood's clumping up, what is that telling us? That's telling us that whatever the antiserum is for whatever kind of cell surface protein, that the blood has that kind of cell surface protein. Okay, is that good? So here, let's say we have a whole bunch of antisera. We have, well, not a whole bunch of kinds. We have two different kinds. We have anti-M serum. We have anti-N serum. We expose blood. This is individual 1, individual 2, individual 3 on a petri dish. We then look for agglutination. Where do we see in this first row? Where are we seeing agglutination happening? Anti-M. Anti-M. Therefore, what kind of blood type is this person going to be? How many say M? How many say N? How many say MN? Okay, how about on the second one? MN. On the third one? N. Okay, does everybody see this? This was a technique. Should we see if you got it right? Oh, look at that. You got it right. All right, we're off to a good start this week. M, MN, N. This is codominant, right? These individuals have a homozygous M, as in Marshall type. These individuals have a homozygous N, as in Nancy type, right? The way that we identify these alleles comes from the guy that developed this test, Carl Landsteiner, and so we have this L, M, M, N, N. Okay, they're codominant. So on the codominant, this is the one, this middle one's the one we want to focus on. This is the one that's illustrating the codominance. Okay, both of the alleles are being expressed. Both of those cell surface proteins are being expressed. Okay, let's look at the exception to the rule of a two allele system. And this actually is more common than not. Most of the time we have lots and lots of alleles at a single gene. So there's a way that we look at that. If we were to look at the existence of any kind of allele, regardless of the frequency in the population, we would probably say that all genes have multiple alleles. All genes have multiple alleles. Because of that, we usually give a cutoff of about 1%. If an allele is lower than 1%, we just kind of ignore it. It's this rare allele, comes into existence, goes out of existence, a new one comes in, goes out. We really don't count things as separate alleles until they get above that threshold of about 1%. Okay, so in natural populations, we call the most common allele in nature the wild type, and as we've seen with Drosophila, we usually denote the wild type by a positive sign. Okay? Other alleles we consider mutant. One thing that students sometimes confuse is that wild types, for some reason, they assume that wild types are always dominant, right? Or the most frequent one is always dominant. It doesn't have anything to do with the dominant recessive characteristic. You can have dominant genes that are in very low frequencies. For instance, if you remember in chapter 3, we talked about some dominant human conditions. One was polydactyly, right? 6-fingered. That's a dominant condition. Is that more frequent than the 10-digit condition? No, even though it's recessive. So what does that do? What is the dominant recessive really doing? Does it make it, is it, if something's dominant, is it necessarily favored? No, not necessarily. Those two things are completely decoupled. You can have something that is recessive and favored. You can have something that is dominant and not favored. Essentially, the only thing that the dominant recessive is doing is exposing it to natural selection. Dominant alleles get exposed much more readily to natural selection. And so if it's something that isn't favored and it's dominant, it might be in low frequency because every time it occurs, it kind of there's selective pressure against it. And so you need to kind of think about that. That's one thing that a lot of students don't quite, for some reason, we sort of assume that wild type is most frequent. Well, not that it's the most frequent, but that the most frequent phenotype is usually dominant, and that's not necessarily the case. Okay, what happens when we have multiple alleles in a single gene? Is anybody in here ever raised rabbits? Pet rabbits? All right. Yeah, there's always. What kind of rabbits were they?
Rexes.
What was their? What did their fur look like? Mini Rexes is what they're called. Oh.
They have red fur.
Is that the one there's a Goliath Rex or not? Oh. What did their fur look like? Anybody else raise rabbits? No. Okay, so sorry, you're gonna be our person. So you're a resident expert. So what kind of fur did they have? Short. What color? Were they all the same color?
Red or white.
So red and white. Okay. So there's a lot of different genes that control the color of fur in rabbits. At most of those genes, there's a lot of different alleles too. So you didn't have any chinchillas or Himalayan? Okay, so never mind, you're no longer a resident expert. On this case, we may get back to you on another one. But on this one, so there is a particular gene that codes for these different kinds of fur colors in rabbits. You have up at the top albino, right? Basically no pigment. You have down here at the bottom wild type, which is kind of this brown. And then you have a couple of other kinds. You have a Himalayan, which has white fur, but then has dark ears and feet and paws, basically. And then you have a chinchilla, which is white hair with black tips, and they're distributed all over the body. Okay, so here we have one gene, but now rather than just having two alleles, we have how many alleles? 1, 2, 3, 4. In the parental true breeding lines, we have 4 different phenotypes also. So it's getting a little bit more complicated. If we wanted to look at the interactions between those alleles, how they behaved with each other, and you had one pen in your backyard with all albinos, another one with Himalayas, another one with chinchilla, another one with wild type, how could we determine what we call the dominance hierarchy? How can we determine which genes influence which other genes, or which alleles influence the other alleles? How can we describe the interaction of the two? What's Cora saying? Crossbreeding, right? Remember that we've come up against this before. Whenever we're not quite sure what to do, just let them mate, right? We like to cross-breed them because then it'll begin to tell us. So how would you do it? Which one would you cross-breed? Albino with wild type. You basically try to combine all of the different combinations. And then you would look for the different phenotypes. So let's say we did that. Here, this is what results in the crossbreeding. So here are the genotypes. So this is obviously, this top one right here is a cross between a wild type and an albino, right? A wild type of chinchilla, a wild type and a Himalayan. What is that telling us? This is the phenotype. What does the phenotype come out?
Wild type.
Wild type. So what does that tell us?
Wild type is dominant.
Wild type is dominant over all three of those other alleles, right? Here we have down here Himalayan versus crossed with an albino. What's the phenotype here? Himalayan. So that tells us that Himalayan is dominant over albino, right? What do the middle two tell us? Do we get some new phenotypes out of here? Yeah, so here's chinchilla versus albino, and you get a completely chinchilla one out of there? No, it's kind of a light chinchilla, right? Here you have Himalayan and chinchilla. What do you get out of it? Kind of a light chinchilla with light Himalayan, right? So what are the interactions? Could you describe these interactions?
Yes.
How would you do it? What's the most dominant?
Wild type.
Then what?
Chinchilla.
I say Himalaya, I see chinchilla. All right. Argue your point. Pat, why do you say Himalaya? Oh, Dan, why do you say Himalaya? You did say chinchilla? No, you said chinchilla.
I said chinchilla.
Oh, Maya said it. Why do you say Himalaya, Maya? Anybody else say Himalaya? Nobody? You're leaving Maya out there? Oh, Travis. Yeah, good. Somebody's got your back, Maya, right? So the Himalayan, why it's completely dominant over this one? Those who say chinchilla, why did you say chinchilla?
I took a stab, to be completely honest.
All right, who's going to help Dan out? Somebody else said chinchilla, I know. So, okay, so let's say we're trying to decide between Himalaya and chinchilla. Those two breed. What do we get out?
Both.
Uh-oh. So there maybe is like maybe a codominance or an incomplete dominance. We don't know the mechanism yet. Okay, but how about chinchilla with the albino? At least chinchilla is way more dominant than albino, right? No, not really, right? So what we see here is a much more complicated scenario, right? We don't have just this dominant recessive. We have dominant recessive going on here. We have dominant recessive going on here. We have some sort of intermediate going on here. And so as we add more alleles, we can see that it gets more and more complicated. Okay, so one great system to illustrate a lot of these things is another human blood type system, the ABO blood type. In this system, we have how many alleles? 4. Somebody says 4 alleles, 3 alleles. Did I say 2 alleles? Yeah, we have 3 alleles. So we have multiple alleles, right? What is the interaction between the alleles? So we have A, B. We have representatives here in class, right? A blood type, B blood type, AB blood type, and O blood type. So we have 4 different phenotypes. A and B, is A dominant over B? Or is B dominant over A? How do they interact?
Codominant.
They're codominant, right? A, B. Is A dominant over O? Here are the different blood types. So if you're AA or A, this is basically, you could say AO, you're A. If you're OO, you're O. If you're AB, you're AB. And so here in this system, again, it's not this straightforward thing. We have some alleles that are completely dominant over the other alleles, right? The A's and A and B over O, and then we have some that are codominant. And we know that these are genes that code for what? What product, Elizabeth?
Surface protein.
Yeah, cell surface protein. So basically, A codes for a cell surface, we've been So A codes for cells in your body that look like this, right? They all have a bunch of A's on the cell surface. B's have this, a bunch of B's on the cell surface, right? And then what do O's look like? They don't have O's, they just have nothing on there, right? So basically it's some sort of mutation that makes it so that they don't produce anything, right? Some sort of, no, I'm not going to go back. Some sort of null mutation, okay? It's not producing anything. The hypo, what was the hypo? It doesn't produce very much. We'll get into those in a little bit. Okay, so we have three different alleles, a null sort of allele, a B allele, and A allele, right? We have dominant recessive, and we have codominant, all within this one system. So it begins to get more complicated. Okay, could you do problems with this? Yes, you could say, okay, which blood type is the universal donor? Okay, think about cell surface proteins. What does Elizabeth say?
Oh.
O, why?
Because it doesn't have any antigen.
Yeah, there's nothing on the cell surface that an antibody is going to be able to move on to. So who's O? Video O in here? Yeah, so if you look around, they seem to be the more altruistic kind of people, right? So which one is the universal receiver? Anybody AB? I think I saw somebody who was AB. Corey, weren't you AB? Who's AB? Okay, no, I'll say I'm AB. I fit the type Y because I'm super selfish, right? I'm the universal receiver. Why?
Because you have all the antigens in your blood cells.
I have all the, okay, yeah, I have all the markers, therefore am I going to make the antigens? No. So I wouldn't make
You have the
antigens.
antigens, you won't make the antibodies.
Oh, yeah, that's what I meant, sorry. Yeah, antibodies. Do I have the antibodies? No, I'm not going to make the antibodies. Yeah, sorry.
You can donate your plasma.
What's that?
You can donate your plasma and then instead of your red blood cells.
Yeah. Oh, okay, yeah, so there's the saving grace. Okay, so ABO type, blood type, multiple, what's the take-home message from this? Complicated system, multiple alleles, right? Complex, not complex, but more complex interactions, okay? This is an example of that. So there are some problems that you'll see in the back of the book where it says, you know, somebody with lady with A type blood mates with a guy with B type blood, and the type or O type blood or something, and then they have a kid with AB type blood. Could the male be the potential father? Stuff like that. You'll see some problems like that. And so you have to. This is one of those systems. It's a simple system. We see it enough. You have to kind of memorize this system. Okay. Okay. So how do we assign a new mutation to a given gene? So say a new mutation arises. When we have a new mutation, essentially what are we generating? A new what? Allele, right? So let's say we have, again, let's go back to rabbits. We have a whole bunch of rabbits. We know that there are a bunch of different genes that determine coat color. If you have mutation in those genes, it makes it, determining or creating this phenotype of coat color involves a bunch of different genes. It's, maybe fairly complicated. All of those genes have to make some potential product, right? If any of those genes get mutated and let's say they're all enzymes and one of those enzymes isn't there, then you're not able to produce fur color, okay? Let's say, again, in our backyard, we know this, all of a sudden we have a new mutation arise that generates a new allele, and that new allele is an albino rabbit. What's going on here? How could we determine which gene that pertains to? So here, this is just this setup. Here we have a whole bunch of different, we have A, B, C, D, right? We know that these 4 genes contribute to fur coat color. We know that if we're homozygous mutant at any of these genes, that the coat color will be white. So 4 independent genes. If you are mutant at any of those, you'll have white fur. We now have this new one. We're just going to call this new mutant C asterisk. Okay. we have this C asterisk, even though, new mutants aren't necessarily going to generate at both of the alleles, right? Let's just forget about that complication, right? If we wanted to test which of these genes that new allele belong to, what could we do? We could just go through and we could mate this one with one of them from the lines of the little a mutants, one of them with the lines of little b, little d, little d. If we do that mating, and based on this nomenclature, we're going to know that it belongs to this C1, right? What kind of organisms or what kind of rabbits should result in that cross? If we crossed a C asterisk with a little a, and they were alleles at different genes, what kind of phenotype would result? Should we draw it out? This is a good place where we start to kind of go back and think, okay, what's happening at the chromosomal level? So let's say here we have, we'll just draw the gamete, right? For the C asterisk individual, we'll say, okay, here's the C asterisk, right? That's a mutant there. That mutation has caused the rabbit to be white, right? Here's another one. It's going to mate with an allele at a different gene. We'll say that gene is down here at little a. We know that this one's white, this one's white when it's in the homozygous form, right? You switch. If those two get together, what is the phenotype going to be? What would you say? Let's first say, okay, let's put more information on this one, on this A. So if it's little a here, what is it going to be here? This is this gene, right? It's going to be wild type, right? If this is the new mutation, right? In that line, it was wild type in the pen, and then all of a sudden, this new mutation occurs, this C asterisk. What is the allele going to be there at the A gene? We'll call it wild type or big A, right? Here's your wild type. Okay. We'll say wild type big A, wild type big C. If those two get together, what is the phenotype going to be? Say this was the A, this was the sperm, right? What is it going to be? What is the genotype going to be? What's the genotype at this allele? We'll call it big C, even not to confuse it with the other one, but big C, we'll say big C asterisk. It's going to be big C asterisk, right? We'll say big C wild type, or big C asterisk, little c asterisk. What's it going to be at this one? Big A wild type, right? Little a heterozygous. What is the phenotype going to be? Let's think about it. It has to be able to produce all four of these enzymes, right? So can this individual produce the big C asterisk enzyme? Yeah. Can it produce the big A asterisk enzyme? Yeah. Therefore, we know it's going to be what phenotype? Wild type fur. It's going to have the wild type fur. If it has the wild type fur, that tells you what? That this is what's happening. Therefore, that new allele is not at the same gene. It's not a new mutation at the big A gene. Okay? If you made here with the B, same thing, wild type. If you made this one with the little C asterisk homozygous with a little d homozygous, and you get a wild type, what does that tell you? It's not there. It's not that new mutation is not of that gene. Therefore, what would you expect here with the little c asterisk homozygous mating with the little c homozygous? What kind of phenotype would you expect? You guys were doing so good at the beginning of class. I'm not doing. Okay, you would expect the mutant, therefore it would be white. You'd have the albino come out. So draw these out. That's just in a classical transmission genetics way how to determine what a new mutant allele, which gene a new mutant allele belongs to. Okay? Does everybody see that? It's looking a little fuzzy, so maybe we'll do another one. Okay, so let's look at Drosophila. We're all, semi-Drosophila experts now. If we were looking at eye color, we know that there are two different mutants. So that was the first one was just a theoretical sort of example. This is a real example. We know that there is a mutation called cinnabar mutation that gives the flies that are homozygous for that cinnabar mutation have bright red eyes rather than the wild type eyes. What do the wild type eyes look like? How many of you have seen wild type eyes? Everybody, right? What does it look like? Kind of reddish, right? Okay, if you are homozygous for the cinnabar, you have bright red eyes. If you're homozygous for the scarlet, you have bright red eyes, right? If we wanted to know if those mutations were the same gene, what would we do? What do we do when in doubt? You made them, right? So cinnabar and scarlet, we cross cinnabar with scarlet. The F1s from that are wild type eyes. What does that tell us? Scarlet's dominant. Anybody agree with that? Oh, Corey, you're on a roll. It's all right though. It's just important to try. All right. So, any other suggestions? They're on different genes, right? Should we draw this one out? Let's just say it's on. Maybe it's on the X chromosome. Oh, we'll just say it's on the number 2 chromosome or something like that. You have here is an individual. Let's say here is where the scarlet gene is. Here's where the scarlet gene is, right? Here is where the cinnabar gene is, right? If an individual is I'm just going to put an little s, right? And here, big C, with the other one, right? What is that individual? What phenotype is that individual? That's a scarlet individual right here. What kind of eyes does it have? Bright eyes, right? Because it's not producing whatever that allele is for the scarlet gene. So here, little s, so this, we'll even draw it out like we used to, big C, little s, is scarlet. Okay, has everybody got that? If an individual is little c, big S, what is its phenotype? Yeah, I heard it. Cinnabar, right? So this one, cinnabar. What's the phenotype? Cinnabar means it has bright red eyes. Cinnabar. Okay, let's go back. Maybe you guys are, but can you see where I'm drawing it on the chromosomes, thinking about it on the chromosomes? And now we're back to kind of doing those dihybrid crosses with symbols. Usually students are more comfortable with those, with the symbols over there than actually thinking about it on the chromosomes, but I want you to be able to kind of toggle back and forth between those. Okay, so here we're crossing cinnabar with scarlet, right? So cinnabar is little c, big S, crossed with scarlet, which is big C, little s. If you cross those, what's the genotype going to be? Good. See, Corey, look how quickly you redeemed yourself. Big C, big S, right? So is it making the gene product at C that it has to have for wild type normal eyes? Yeah. Is it producing the gene product at S that it has to have? Yeah. And so this is wild type. Okay. Because you have wild type, because you get a wild type phenotype out of there, it's telling us it's 2 different genes, right? If you were to cross this new mutation, we call it cinnabar, or cinnabar 2 with scarlet, you get a wild type out, okay? But if we were to cross cinnabar 2 and cinnabar, we get a cinnabar or bright red eye resulting offspring. What does that tell us? You're rolling. You want to answer. What does that tell us? Are they different genes? No, they're what? The same gene. Okay, that's exactly what that's telling us. This is, in a practical sense, how we're going to assign new mutations or new alleles to the different genes. Okay, and it gets complicated when you have. So here we're going to show you. There it is. Cinnabar 2 and cinnabar are alleles of the same genes. These are different genes. These are different genes. Okay? It gets complicated when we have phenotypes that it's hard to distinguish between. But this is where, in the early days of genetics, this is what they had to do. This is in Morgan's lab, fly lab. They had to try to figure out all these things by just doing all of these different kinds of crosses.
So we're not
Tyler?
seeing all of the offspring are mutants. We're just seeing some of the offspring have the mutant trait.
No, well, so in this case, so say you're true breeding cinnabar, right? Let's just make it simple and say you're true breeding cinnabar 2, right? So here we go. We'll just draw out how we know it already. So we'll just call it C2, C2. Those have cinnabar eyes, right? We have ones that are here, little c, right? What are all of the offspring going to be? True breeding here, cinnabar, true breeding cinnabar 2. The resultant offspring is going to be C2, little c2, c. Neither of those make the requisite gene product to have normal eyes, and so all the offspring are going to have here. Okay. All right, does that make sense? Basically, just kind of think of it in the beginning, before drawing it out here on the chromosomes, just think, okay, if you have a new mutant and you cross it with an individual and you get a wild type out, and you get a wild type offspring from that cross, what does that tell you? Different genes. If you get a mutant phenotype out of it, what does that tell you? The same gene. So just first maybe memorize that and then go and sort of go back through and draw it out on the chromosomes, and then it'll make a lot more sense. Okay. So as you can see, we're getting into more and more complicated scenarios. Okay, so as we get into these more complicated scenarios, we have to now begin to describe and maybe even name different kinds of mutations. And so when we talk about visible mutations, it's pretty straightforward. We're basically talking about a mutation that has some sort of effect, some sort of visible effect on the morphology. We'll talk about sterile mutations. These are mutations where they limit the fecundity or the reproductive success of either the male or the female. And the lethal mutations, pretty straightforward too. These are mutations that occur that are that affect some sort of vital function in the organism. Therefore, if you have the mutation, you're not going to survive. Okay, so these are just some broad categories of mutations. Okay, let's go back and do some crosses. Let's say again, not to beat a dead horse, but let's say again, you have a population of mice. This agouti mice, this is kind of the wild type fur color of mice. And then let's say all of a sudden okay, does anybody have pet mice? No? Maya, no pet mice ever?
Take care of mice.
Okay, but you never had your own. We'll just use you as the mice keeper. Okay, so Maya keeps mice. Let's say Maya has a whole bunch of wild type mice. Carl, you have mice to feed your snake, huh? Yeah. Oh, and you do? Okay, so never mind Maya. We're going to go with Carl. Carl has mice. They're wild type. Let's just pretend they're all these wild type. This kind of fur that we call agouti fur is this sort of gray-brown wild fur. Then all of a sudden Carl goes in, decides he needs to feed a snake. He goes into his mice population, and he sees this new yellow kind of chunky mouse. He's excited, right? Why? It's nice and chunky, so it'll be good food for his snake, right? But then he says, hey, this is interesting. All right, I have this new mutant. to do some experiments. What do I want to do? I want to figure out, because when a mutation first comes out, what do you want to do? You want to kind of describe what the mutation, you want to figure out how this mutation is interacting. You want to figure out how it interacts with the other alleles, right? You want to know if it's dominant or recessive. You want to know all of these different things. And so what do? When you want to try to figure out stuff? You breed it, right? And so you say, so Carl takes this yellow mouse and he mates it with this agouti mouse and he gets the offspring, and what he sees is maybe he breeds 20 of them, 9 of them are wild type and 11 are yellow. So roughly a 1-to-1 ratio. What does that tell us? So are we already beginning to be able to describe the interaction? What does that tell us about that new mutation? Okay, so Carl says codominant. Any other ideas? What do you think? Codominant. Recessive? No? Okay. So, should we draw it up again? So when in first out you make them, and then when you're in doubt even after you make them, what do you have to do? You have to draw it out. Try to make some sense out of it, right? Okay, so here, in this case, you have a whole bunch of mice, right? A whole bunch of, what should we call them? We'll call them wild type mice, right? Wild type, well, no, that's going to be hard. We'll call it big A. Big A mice. So here, I'm just going to draw maybe 5 individuals, right? Big A, 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 big A. What happens at some point in time before the night, not the night before, but when they were breeding, before Carl came in and discovered it, you had, what's this? Mutation, right? So one of those alleles gets mutated. This new allele, this new mutation, this just usually happens just in one of the alleles, right? You don't have mutations that usually change both of the alleles, but rather it just changes one of the alleles. Okay, so now the new mutation, we'll just call it AY. Okay, that's the yellow one. Even though that this isn't following our typical nomenclature. Okay, so now we have this yellow one. We made this yellow one, right? Now with an agouti, normal one, we do the Punnett square for that. Big A, big A, big A, Y. What do we get? Big A, big A, big A, big A, big A, Y, big A, Y. What's this ratio of genotypes? 1-to-1. What are the phenotypes then? Baby yellow agouti, only if what? It makes sense here and it makes sense down here. How is the yellow interacting with the agouti? Dominant, good. All right, so we do that one mating and all of a sudden now we say, okay, we're beginning to figure out the interactions because we're looking at these ratios, right? This is the kind of skill set that I want you all to acquire, to be able to say, oh yeah, okay, I see this ratio. Thinking back, creating these Punnett squares in your mind, you say, okay, that makes sense. One, you have all of a sudden a mutation occur, right? And maybe this mutation occurred a couple of generations before as a recessive. It kind of got passed down to a couple of other ones, but you couldn't see it, right? And then all of a sudden, maybe first cousins got together. You had the 2 recessive alleles come together, and then all of a sudden it popped up, right? That could have been one option. But now, if that would have been the option, right, and you would have mated it against an agouti, what would you have gotten in your F1s? They'd all be agouti. Good, Carl. They'd all be wild type. So now we're distinguishing between those 2 scenarios. Now this is telling us, okay, the yellow is dominant over the agouti, and we have it in a 1-to-1 ratio. Okay, good. So look at all this stuff that we can actually figure out just by kind of working our way backwards. This is classical genetics, all right? Okay, so now what are we going to do next? We decide, okay, that's cool. We figured out it's dominant and recessive. But let's try to figure out what else is going on. What should we do? Let's hear it all together. Mate, right? All right, we want to mate. So then we say, okay, now that we have a bunch of different agouti, or a bunch of different yellow ones, let's let the yellow ones mate with each other. And so we have the yellow ones mate with each other, and you get a clutch size of 15, right? Of that, you get 5 agouti and 10 yellow. Now it's getting even more complicated, right? It's like, okay, what are the ratios here? Can we figure out these ratios? Yeah, so here we see, it's 2 to 1. Let's say we say, okay, 2 to 1, but is that close enough to 3 to 1? Because we're used to that. We're comfortable with that 3 to 1 ratio, right? So we have this unconscious bias to make it easy, right? So we're saying, okay, 3 to 1. How would we decide if that's close enough to 3 to 1?
Chi-squared.
Yeah, we do a chi-squared, right? And let's say we do it and figure out, oh, unfortunately, no, that's not close enough to a 3 to 1. So it's a 2 to 1 ratio. Have we seen a 2 to 1 ratio yet? So it's a 2 to 1 ratio. So now this is a different ratio, right? 2 to 1 ratio. We're pretty sure it's 1 gene. We can actually draw out. We kind of think we know those genotypes. So let's say, okay, we got the ratio. We're having trouble. Let's try to draw it out on a Punnett square. Here we go. We draw out the Punnett square. Big A, big A, big A, Y, big A, Y, Y, Y. This ratio should be what? If yellow is dominant, it should be 3 to 1, but we're not getting that 3 to 1, right? We're getting the 2 to 1. Okay. So what's going on here? Any of you? What's that? Corey, did you say something? Who? Tyler, did you say something? Who said something? Okay, so now we got to go beyond. This is kind of the critical thinking part of genetics. You got to start to be able to say, okay, I got to try to figure this out. We're getting beyond just like memorizing facts, right? Memorizing things. We got to try to figure it out. Can anybody figure out how we could get a 2 to 1 ratio out of here? What car? The homozygous yellow. All right, so yeah, good, Elizabeth. So let's say we do this. We just cross that out. What does that give us? 2 to 1. A nice 2 to 1 ratio. And sure enough, with the yellow and agouti, that's exactly what happens. And so here they've done it differently. They did A with the Y and the wild type, but we understand it, right? Here we have our yellow, and our yellow we made agouti, yellow, yellow. And what we see is that not only now are we describing the yellow mutation, not only can we say, yes, the yellow mutation is dominant, but now we can also say in the homozygous condition, it's also what kind of mutation? We described 2 slides ago? It's a lethal mutation. So again, just by mating, we're beginning to describe more and more about this new allele. Okay, so it's dominant and lethal. Yeah, and so Carl said, so a lethal mutation doesn't necessarily kill all of the offspring. For instance, if it's heterozygous, in this case, it didn't kill it at all, right? And could there be lethal mutations that do that do kill all the offspring, even in the heterozygous state? Yeah. Could there be cases when it doesn't? No. Could it depend, maybe? We talked about this, I think, the first week. Could it depend on the environment too? We talked about an example when the expression of that when the expression of the heterozygous and the homozygous mutation depended on the environment. That was sickle cell. Remember sickle cell? So sickle cell heterozygous in a non-malaria environment, maybe would be considered more lethal or more deleterious than in a high malaria environment. Yeah, and so it's getting more and more complicated. Not only is it getting complicated depending on how these alleles are interacting, but it's going to get complicated, and we're going to deal more with that complication of the environment in a little while. And so things are getting complicated, and again, thinking back to why it took us so long to figure out all this stuff out, these are the reasons why. Because scientists were trying to figure it all out kind of at once. And it wasn't until Mendel came along and said, all right, let's just get a really, really simple scenario, a really, really simple experiment going so we can at least establish some of these general ideas, and then we'll build on it. And that's exactly what we're doing right now, is we're building on it. Oh yeah, I forgot to change this. So we have just looked at a whole bunch of different cases where we had all these different symbols, right, for the different alleles. There is a convention, before today we sort of tried to follow it, of how there is kind of a convention of how we name alleles, capital versus lowercase, et cetera, on page, I think it's 68 in the new one. This has the old page. I think it's page 68 in chapter 4. There is a focus on genetic symbols that you read, and it kind of explains how we generally name our different alleles. We already kind of know what do we usually call a new mutation, or how do we usually select the letter for a condition, or how have we done it in the past, like I'm talking about albino. We usually use the mutant one, right? Recessive is the lowercase, and that's usually one that we use. We use the first letter of the condition. So, for instance, albino. The recessive, we generally do it as lowercase. But as you can see, what we just barely did, there's a lot of exceptions to that rule. So just read it to know that's generally how it happens. And then as we go through the book, you can count, on not just your hands, but also probably your toes, the times when you're given problems where it doesn't necessarily follow them. A lot of the reason it doesn't usually follow that, or doesn't necessarily follow that, is because, like for instance, with the MN blood type, it predated kind of doing this. And now when we're getting more and more alleles, it gets harder and harder to name those different alleles in a really consistent way. So I just want you to kind of go through and read that just for your own education, kind of. Okay, so let's, what do we have, 5 more minutes? Oh, today went by fast. Let's, in the last little bit, let's go back and try to think about all of these things, think about all these alleles as gene products. So as we go through these next couple of chapters, I want you all, as you do it, to not only think about it in symbolic terms like we've done here, big A, little a, big B, little b, but also in chromosomal terms, where is this gene going to be located on the chromosome? Where big A, little a, but think about it, where its specific location or locus on the chromosome. And then a third way that I also want you to think about it is even at the higher resolution, that is, okay, what are these actual gene products, these sequences of amino acids that are being coded by these alleles? How is that happening? Okay, so here we have a bunch of different genes, right? We have gene A, gene B, gene C, gene D. We can think of them as sequences of nucleotides, right? Within the genome, those sequences of nucleotides get transcribed, and then they get translated into amino acid chains. These amino acid chains are what we call polypeptides, right? So we actually have a protein product of the gene. Here, this is just 4 different kinds, right? These different amino acid chains will affect some aspect of the general phenotype. They could be, like we mentioned before, enzymes that catalyze certain reactions. They can be structural components. They can be proteins that are going to transport pigment to certain parts of the cell or to certain cells. They can have a whole bunch of different functions, right? I want you to begin to start thinking about these gene products in sort of from a cellular functional perspective, okay? So let's just say, let's go back and look at dominant recessive mutations and say, okay, what would the gene products look like in the case of these dominant recessive or loss of function or hypomorphic, like when we looked at the rabbits when they don't have quite, they function a little bit but not quite like they should. What would it look like? And so let's just look at that first gene A, and we'll say, okay, there's a wild type. This is what the wild type allele produces, an amino acid chain, and we're going to give it really simple three-dimensional conformation, right? And it kind of looks like that. That's a function. We'll just call it an enzyme, right? We'll say this is an enzyme. The utility or the function of the enzyme depends on its three-dimensional structure, right? This is what the three-dimensional structure should look like, okay? That is the wild type. Let's say we have a recessive amorphic loss of function allele, right? We can even think it back to rabbit fur color, and we're going to call that little a. When little a produces its amino acid chain, its polypeptide, it looks like this, right? It's all kind of wavy and as a three-dimensional form. Do you think it's going to produce or provide the same functionality as the first one? No, that one doesn't have any functionality. It doesn't form as an enzyme at all. Okay, so if this were our rabbits, it would not produce any sort of pigment, right? It wouldn't have the enzyme at all to produce the pigment. It wouldn't work at all, therefore you'd get a white rabbit. Okay, next one, a recessive hypomorphic loss of function. So it has kind of the shape, right? It's a little bit the shape, but it's not the best shape to do what it's supposed to do. And so it can catalyze every once in a while. It lowers the activation energy a little bit, but not really, let's say, that much in the enzymatic reaction. And so it, it kind of produces a little bit of fur color, but not very much of the pigment or whatever, okay? And then let's say we have a dominant negative one. Okay, so this is what the three-dimensional form of that allele product look like. Not only does it not function, but it interferes with the function of the wild type. And so if you were to go through and cross these, we want to say, okay, let's try to make an allelic series out of it, figure out which one's dominant, recessive, or whatever. Let's go back through. We did it. We're kind of already telling you which. But let's draw out those genotypes like we did before. But now let's also look at what the polypeptides that are present would look like. So here we have a wild type with the A, right? So we have a bunch of good enzymes and we have a bunch of bad enzymes, but we only need a very small amount of these enzymes to get wild type fur. That's what that would look like. That would tell us that that was a recessive one. Because it's an enzyme, we don't need very much. Here, this is a hypomorphic one, so we have a good enzyme that works just fine. We have another enzyme that kind of works, not that well. We breed those two together. The phenotype would be wild type. That tells us it's recessive. Now, the third one, we do the wild type with the dominant. This dominant one down here now interferes, maybe it binds to that wild type enzyme, totally messing up its three-dimensional shape. Therefore, it can no longer function as an enzyme. It therefore gives you a mutant, let's say a white-furred mutant phenotype in the end, and we can now say that that is a dominant recessive type thing. And so a lot of times when we look at the albino or the white fur color, essentially what we're looking at are enzymes that don't work. The amino acid chain doesn't fold up right after it's produced, and so it doesn't function. It's sort of a, I'm not sure what that is. It's sort of a loss of function there. So, loss of function mutations, cinnabar is a loss of function mutation. It doesn't produce the enzyme that is needed to produce that brown pigment in the eye of the fly. That's why it's bright red. And so the normal wild type fly color is a mix of brown pigment and bright red pigment. With the cinnabar, you mutate an enzyme that's necessary to produce the brown pigment, therefore you just have the bright red pigment. That's what gives you that phenotypic difference. There are also such things as gain of function mutation. We mentioned this a couple of weeks ago. And antennapedia. The antennapedia has to do with the developmental genes. Essentially what it does is here is a normal fly with normal antenna looking at it from this perspective. The antennapedia actually makes it so rather than producing an antenna, it produces a leg, two legs in place of the antenna. That's the pedia part, the leg part. That is a gain of function. I don't know, it's not a great function, standing on your head or something, I don't know. But it does have some sort of function, but that's important that we can actually gain functions from mutation. This is especially important when you start to look at evolution and Okay, so for the last minute here, we're going to come back to the rabbits. We're going to come back to the Himalayan mutation, and it is the product of a partially functioning enzyme. Okay, it functions, but it doesn't function that well. I want you to figure out exactly what's going on. Try to figure out between now and either lab or next lecture time what's going on. What's going on with the Himalayan rabbits with the color distribution, and think about it in terms of an enzyme product and what you know about enzymes. Okay? All right, so with that, let's stop, and I'll see you at lab today. We'll look at a whole bunch of more flies. So now we are going to look at gene action. We're going to go from genotype to phenotype, but we're going to do it in a more complex way. This whole chapter, chapter 4, is about basically looking at some of these same things that we looked at in chapter 3, but relaxing assumptions, looking at more complex, complicated scenarios. And so in chapter 3, we essentially said, okay, genotype roughly equals a phenotype. If you have a certain genotype, it's going to equal, there's sort of a one-to-one relationship between that genotype and that phenotype. But we know that phenotypes depend both on the environment and on genetic factors. And so there's this sort of nature-nurture is kind of what it gets played out as in popular press and things. There are both components that determine a phenotype. Genes do not act in isolation. Okay, so let's look at some scenarios, or let's look at some cases where we could potentially have the same genotype, right, but then have different phenotypes because of these environmental causes. And when we talk about environmental causes, we're talking about both biological and physical. And within the biological environment, we might even be talking about the genetic background of a genotype. So here, before now, we've just sort of been looking specifically, okay, one big A, little a, one genotype, that equals this phenotype. But in reality, there's a lot of other things that play into it. And now, we may talk a little bit about it this semester. I'll probably have you read something at least about it. We now are discovering that not only are these things important, but the environment of a parent are also important. This is something that we call epigenetics. So the regulation of these genes can now be passed on, and every year it keeps getting more and more complex, trying to understand this, and each year we discover that it's more and more, the environment of a parent influences the offspring. So for instance, some of the first studies in epigenetics, they altered the diets between clonal mice, so they had exactly the same genotype. One group had a really high fat diet, the other group had a really lean diet, and in fact, when they had offspring, some of the control of different genes were also passed down to where the offspring of the parents, also of the fat line, the offspring of the parental generation that was fed a high-fat diet, the offspring were significantly more fat than the others. So that kind of goes contrary to what what up until about 10 years ago we just thought, no, it's all about the genetics. Does anybody remember Lamarck? They're kind of calling this neo-Lamarckianism. Those of you who've had genetics, what is Lamarck infamous for?
That change occurred within an individual instead of a population.
Yeah, so in acquired characteristics, the inheritance of acquired characteristics, we now know that there are some cases with that. For instance, we're finding out parents that smoke, a child is raised in an environment where there's a lot of secondhand smoke, that alters how not just the child that's raised, but the parent that spends their lifetime smoking, that will alter how their genes are regulated, and then those get passed down to the offspring. And so the types of, you know, mutagens that you're exposed to. So the bad air, the inversion, is not only affecting all of us right now, but it could be affecting, determining some genes that are being turned on and off in your offspring. And so it's kind of a crazy thing. So we'll talk a little bit about that. That's sort of like kind of cutting edge stuff. But before that, we need to kind of look at what are some environmental factors and kind of look at some examples, kind of go establish a foundation to where we get away from thinking about just gene equals phenotype. So we're going to talk about 3 examples, just briefly. One, we're going to talk about Drosophila. There's a mutation in Drosophila that's called Shibari. It's called the Shibari mutation. We'll talk about the environmental influence on the expression of those genes or that genotype there. We're going to talk about phenylketonuria. We talked a little bit about that a couple of lectures ago. And then we're going to talk about pattern baldness. Okay. First one, the Shibari mutation. So flies with this Shibari mutation. Does anybody speak Japanese? No? I think Shibari is like either paralysis or earthquake in Japanese. I can't remember which one. But they were Japanese researchers that discovered it. What they found is that at 25 degrees Celsius, that's about room temperature, somewhere in the 70s Fahrenheit, that this is what a fly with the Shibari mutation looks like. Okay, can you see any sort of visible mutation? You're all experts at identifying Drosophila now. What can you tell me about this fly? Let's see, is it a male or female? Female, good. Look like it has pretty nice eyes. Are those nice, regular Drosophila eyes? They look pretty good to me. How about the wings? Are those like, those look like wild type wings or vestigial wings? Wild type, right. So it looks perfectly normal, okay? This, in fact, a fly that, lives this life in one of our vials, right, at 25 degrees, which is pretty much what we have here, would look like this, go throughout its whole life like this. Nothing would seem phenotypically wrong with that fly. What happens, though, if, we decide to bring it in and do some things in here, we want to transfer it to the another vial, we kind of shake, we can shake the vial up. When we are shaking the vial up, what are we doing? We're getting ready to transfer it, right? But what are we putting into that system? What is it? Motion. What's motion? What kind of blank? Kinetic energy. Right, so we're putting kinetic energy into that system. That has the effect of temporary paralysis for the flies. So you shake it up, you have like a little mini earthquake, right? And all of the flies fall to the bottom of the media and are paralyzed. They recover from that. Okay, so they eventually recover from that, and then they, return to being able to fly normally and everything else. If you raise the temperature, so the heating in this building, in this building they wouldn't survive because half the time we're freezing and half the time we're like probably way above 29 degrees. But as soon as we have one of those days where it's really hot in this building, 29 degrees, they not only become paralyzed, but they fall to the bottom of the vial and they die. Okay, so what's the explanation for this? Are you seeing a fly, Cooper? We'll see him for the rest of maybe this week or so. So what's going on here? What's happening? Okay, good. So we sort of led into it, good Carl, with the rabbit example, but it's probably some sort of enzyme, right? Some sort of enzyme that, one, has a dramatic effect when it adds energy to the system and it kind of changes its form. It can't perform its normal function. But as soon as that energy is taken away, right, it kind of resumes its normal three-dimensional structure, and it can, behave as it's supposed to behave. But if we put it at 29 degrees, it's never able to conform back to it, and it ends up dying. Christian?
So the temperature change, I can see where that comes in, but the by shaking or by adding a little kinetic energy, that kind of surprises me. Is that because the enzyme is just really unstable or something?
Yeah, so the enzyme's unstable. You're adding kinetic energy, probably heating up the flies a little bit. So Putting it at 29 degrees just makes a constant higher heat. Shaking it just probably adds a little bit of adds a little bit of heat. I mean, how many of you ever saw, like 10 years ago, maybe more than that, 20 years ago, the little rubber band thing they'd put around and they'd shake your love handles, and it's supposed to get rid of. So you can see, like, when you're adding things and shaking up, you add a little bit of heat too, right? Anybody ever use them? Needless to say, I don't think they work very well. Okay, so yeah, good explanation. But here, for our purposes, what's going on? The genotype isn't changing, right? A fly that is living at 25 degrees compared to one of these flies that's living at 29 degrees, the genotype's the same, right? But what's changing? The environment. What is that having an effect on phenotype? These two phenotypes, dead versus alive, it's a different phenotype, right? Being able to fly or not being able to fly. Another one. We'll just go through these really quickly. So we mentioned PKU earlier. This is a condition. Under normal conditions, you have a phenylalanine hydroxylase, which is an enzyme that converts phenylalanine to tyrosine. Okay, people with PKU are not able to produce that enzyme, the phenylalanine hydroxylase enzyme. And so what happens is you get buildups of phenylalanine. When phenylalanine gets built up into high concentrations, it converts to phenylpyruvate. Normally, this enzyme is going to, crank along and convert phenylalanine to tyrosine. It's not able to do that. You get high levels of phenylpyruvate. If you continue to get high levels of phenylpyruvate, eventually you will develop problems in your brain development, progressive mental retardation, and seizures. So big consequences of this buildup of phenylalanine, phenylalanine that then gets converted to phenylpyruvate. Okay, so what kind of environment, what's the environment here that we're talking about? So if can a person that is not able to produce this enzyme have a completely, we'll say, quote unquote, normal phenotype?
Yes.
So Dan says yes. So what is it dependent on then? Diet, right? So the exposure to phenylalanine. And so this is the reason why when you have a newborn baby, I think they pretty much just do it anyway, but especially if the baby comes out and it has kind of this musky odor to the sweat, they will for sure do a test for phenylalanine. If you, what's that? It's required. Yeah, I mean it's one of those things you might as well just do it, right? And so if it comes back that the infant does have PKU, what then does the parent do? Alters the environment, right? Alters the environment so that the baby is not exposed to phenylalanine. What were you going to say? I'm sure there's full. How many of you had an offspring recently? What are the tests that they do? Remember? But there's a whole bunch of them. Because you might as well, right? You might as well do it because it's not an easy fix, but it's an easier fix than dealing with the disease, than dealing with the problems, right? And so here you have formula that is produced that doesn't have phenylalanine in it. Aspartame is something that it's not as much in our foods, in our diet foods, especially now because we have Splenda. So Splenda is probably just as popular, but it used to be that pretty much all of our diet foods, at least here in the U.S., had aspartame. In other parts of the world, they had a lot of saccharin. So how many of you have ever had sodas in different parts of the world that taste different, like Mexico and stuff? That's because they have saccharin in them. And so originally saccharin was developed as a sugar substitute, but then there were a couple of studies that they gave massive doses to mice, and then it caused cancer. And so then we freaked out here in the U.S., and we banned it all, which probably was an overreaction to it. But that's, it was sort of this cultural inertia, and so then we started with aspartame, and aspartame is probably, we're finding out, is probably even worse now because it breaks down into some, formaldehyde and a whole bunch of other stuff. But not only that, for people with PKU, one of the parts of aspartame is phenylalanine. So if you're PKU, you definitely want to avoid any of these kind of sugar substitutes. Okay. Next one, male pattern baldness. So this is a condition that oftentimes in popular culture or just in the general public think that it gets mistakenly viewed as a sex-linked trait. It is not a sex-linked trait. Here in females, and we'll draw them up here. Let's see. So we'll just call it big B or something. So in we'll do the females. So in females, you have different genotypes, right? So we'll just say little b, big B, little b, big B, big B. In females, you have to be heterozygous, or homozygous rather, for this condition to have pattern baldness. And even then, the phenotype isn't as severe in males. And so in females are bald if they're homozygous recessive, or homozygous for it. So this kind of seems like it's a recessive condition, right? In males, on the other hand, and then we'll just, don't get confused, I'm just going to do this part. Well, I'll erase it. In males, if you're homozygous or heterozygous for it, so it kind of seems like it's a dominant characteristic. So males that are, we'll just say BB or BB, little b. So just to be consistent, we'll say, okay, females here, if they have it, males, if they have big B, or big B, little b, they develop male pattern baldness or pattern baldness, and it's even more extreme, the actual phenotype. So what's going on here? And so some of you, how many of you have heard like, well, if you're bald, it depends on your mother's father or something like that, or all of these different things. It confuses people because they think, okay, it is sex-linked, but it's not sex-linked. Okay, what is it? It's environment dependent. So what's the environment that we're talking about here? Think of the biological environment here. Hormones, okay, so what's the difference between males and females from a hormonal perspective? Males have high levels of what? Testosterone. And so here you have a phenotype that the expression of that phenotype is dependent on the biological environment. And so in an environment where there is a lot of testosterone, you have a certain phenotype. And so even the heterozygous here shows this pattern baldness. Homozygous does here also, but in that high levels of testosterone. So how does that kind of seem like it's sex-linked? Where's the gene that we have that produces testosterone? We'll get into this in, I think, next chapter a little bit more. Does anybody know? Where would it make sense that the gene is? On a Y chromosome, right? Right, but what we're going to see is it's actually a little bit more complicated because on the X chromosome, there's another gene that has to be produced, has to be expressed for cells to be receptive to testosterone and the changes there. So it actually, when we talk about gender and stuff, it gets a little bit more complicated. But again, here's another example of a genotype being dependent on an environment. Okay, how else can environment, what other things happen in the different environments? We noticed when we started doing pedigrees, we started looking at a lot of pedigrees, we noticed that sometimes some abnormal behavior in the pedigrees popped up. We found out that sometimes, even though an individual, based on pure genotype, should express the condition, sometimes they did. This is the case of incomplete penetrance. One example of that is polydactyly, that's six fingers, right? If you remember back to the slide a while ago, this was one of the characters that we listed in human genetics that we know is a dominant character. But we see that sometimes the individuals that actually have that genotype don't necessarily express that phenotype. If we were to look at that in a pedigree, we could identify it once we know what the normal behavior of dominant and recessive traits are in a genotype or in a pedigree. Here, could you identify the place where incomplete penetrance is occurring? So if you looked at this just in the beginning, you would say, okay, this character, this trait looks like, would you say dominant or recessive? Dominant, right? Because you have here parent, 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 all the way through every generation. But then if you were to only look at this part, what does this part kind of look like? Recessive. So people were saying, oh, what's going on here? That seems kind of weird. What we now know is that in fact this individual has the allele but does not express it. This is what we call incomplete penetrance. And certain conditions are more prone to this, or certain genotypes are more prone to this than others. Pat? Autism's complex. So autism's been in the news lately. Why? Does anybody know? With the measles and everything, and the supposed link to vaccines and all that. So autism is complex, and it seems to be a condition that can come from a lot of different things. And so there's no gene for autism or no autism. There may be a bunch of different genes for a bunch of different things that eventually causes children to develop these autism-like symptoms and stuff like that. But we're not anywhere close to being able to draw out autism on a pedigree. So yeah. So the answer probably, I would say, probably no with autism. Autism is just too complex. It's too complex in the sense that we could be piling in a whole bunch of different causes into one sort of syndrome. You know what I mean? Okay.
So, much emotional. I had expressed polydactyly. I had that removed when I was about 3. So far.
Oh, really?
Yes.
Oh, that's cool. You're the first student I've ever had that had that. So expressed in just one hand.
Yeah. I just had an extra thumb. I had a fingernail and everything.
Oh, wow.
I had it removed when I was 3. They wanted to wait for the nerves to kind of form before they operated on it because it could have paralyzed my actual thumb.
Oh, wow. Yeah. So it's a dominant. So because you have you been genotyped for it then?
I have not.
And so you just had a kid, right? So
far he's not.
So just one kid. Yeah. So that'd be an interesting thing. So now this class you just saved a genetic counselor fee, right? Because now you kind of know. Okay, so what's going on? But as somebody who didn't know, it'd be like, oh, what's going on with this? Why is this? So now we know that it is a dominant trait. So there is a possibility that what's the chance that your offspring is going to have polydactyly? Well, have the genotype for polydactyly, right? 50-50, right? But then that also, what happens with incomplete penetrance, sometimes it depends on your other genetic background, right? And so you are probably also contributing those alleles. So I would say, and I don't know exactly if it's the incomplete penetrance on polydactyly is because of your genetic background or environmental background. Maybe a little of both, but you probably have a substantially less than 50% chance of giving it to your offspring. So that's interesting. So in your first offspring was it apparent at birth, right?
Yes.
Yeah. So for sure your offspring. Oh, that's cool. Yeah. All right. Okay. So incomplete penetrance right here. You'll have to keep me. When you have other kids, you have to keep me informed. I have a great example. That's awesome. Okay. Complete penetrance. Okay, another something else that we see is this term that we call variable expressivity. And so here, each of these flies has I think it's a homozygous recessive condition called lobi mutation in Drosophila. Each one of these flies has exactly the same genotype. But if we look at it, basically what it causes is, the eye to form this sort of lobe shape. But if we look at it, we have on this end a fly that kind of looks perfectly normal eye. On this end, a fly that doesn't have any eye at all, and we have basically everything in between there. And so these are genetically identical, or the genotypically identical, but then there is variation in how they're expressed. So it continues to get more and more complex. This is something we call variable expressivity. The variable expressivity could be due to environmental factors, could be due to biological factors in a developing embryo. It could be due to genetic background. And so there's all of these things that are at play with the expression of genotype. Okay, I mentioned genetic background. We know specific situations when not just the sort of general genetic background influences the expression of a particular trait, but the genotype at other genes influences it. And so you have a lot of genetic interaction going on. In the early days, shortly after the rediscovery of Mendel's papers, the turn of the century, Bateson and Punnett, who were two. Do you remember I mentioned De Vries earlier? Those were three of the people that were really influential in establishing genetics as a science, and they were the ones that were responsible for rediscovering Mendel's work. They read Mendel's work. They maybe read De Vries' work, and they said, "Oh, why don't we try to figure something like this out on an animal system, right? Because De Vries was working with campions, Mendel with peas. Let's see what's going on with animals." And so they decided to experiment with chickens. Okay, anybody raise chickens in here? All right. So with eggs, you guys do eggs? Really? Oh, cool. I used to have a neighbor that used to have them. Now she doesn't do them anymore. I love homegrown eggs. Okay, so here are a couple of different kinds of chickens. The difference between these chickens are their crests. We have different types of crest. This is called a rose crest, right? This is called a pea crest for obvious reasons. Kind of looks like a row of peas. This is called a walnut crest, kind of looks like a walnut. And this is called a single or a leghorn crest. This is kind of like the, oh, it didn't pop up here. Did I take it out? Oh no, there it is. Foghorn, leghorn, that's where he gets his name from. Do you ever remember that cartoon? Okay, yeah, so you have four different morphologies for the crest. And so they said, okay, let's take a true breeding parental line of rose crest, a true breeding parental line of pea crest, and let's do kind of like what Mendel did, right? And he crossed them, all right? So what do you think they were expecting out of this? Let's say they said, oh, we're gonna make it really simple. We're just gonna use rose and pea chickens. What did they expect to come out of there? Either a rose or a pea that would be dominant or recessive. But what ended up coming out of there are walnut. All of the F1s were walnut. So they're a little bit confused. They're like, a new phenotype is kind of weird. But when in doubt, what do? Just let them mate, right? So they let all the F1s mate with each other. He said, okay, this is going to be interesting. What's going to come out of this? All of a sudden now, in that F2 generation, they got pea chickens out of there. They got rose chickens out of there. They got walnut chickens out of there. And then they also got an entirely new phenotype out of there, the leghorn phenotype. So they were like, wow, what's going on here? So we're going to pretend we're them. And here are your results. I want you to try to figure out what's going on here. So what are we going to do? This is good exercise for, what you should do when, as young budding geneticists, what do you do? So here we have, these are our results. What are you going to do? You can do it as a table too. So yeah, maybe we'll do it as a table, and then each of you come up, we'll give you 3 minutes in each table. I want to come up with an explanation of what's going on without looking at. Okay, so you guys are getting close. So those are good. So you guys are on the right path, but essentially what do you want to do? You want to look at ratios. You want to try to figure out, okay, what's going on. We're going to do a bunch more with ratios here in a little bit. But then you want to say, okay, that 9:3:3:1 ratio sort of looks familiar, right? 9:3:3:1, 9 walnut to 3 rows to 3 pea to 1 single or leghorn. What we find out is that it's behaving exactly how our dihybrid cross was behaving when we were doing it before with Mendel's, with the Mendelian system, only rather than having two different characters, right? Well, first we should ask, are the two genes, if we're looking at two genes, are they assorting independently? What do you think, Corey? You're the one that always says independent assortment. All right, yeah. So they are because we got that 9:3:3:1. They are assorting independently. But what's different, rather than having a combination of characters one gene pertaining to one character, another gene pertaining to another character. So, say for instance, the height of a plant versus the color of the pea flower, right? It's actually both of those genes are influencing a single morphological character. And so we can go back through now and we can look at the parental generation. We say, okay, there's two genes. The parental generations are true breeding, right? And so here we have big R, little p, little p. That big R is a dominant allele. So in this row's lineage, all of the offspring are going to be big R, right? And they are all going to be rows, have the row's crest. In the other one with the P, you have little r, big P. You really, for that phenotype, just need one big P, but because the parental generations are true breeding, they're big P. You put those together, what is the resultant genotype when you cross those two? Big R what? Oh, I think I said heterozygous, somebody said. Big R, big P, little p. And now we know that big R whatever, we can do a dash, big P whatever, gives us what phenotype? What was the F1 phenotypes? Walnut. Yeah, so that's exactly what happens. And so then we take the walnut, right, which is big R, big P, little p. We made those together. We get our 16-cell Punnett square. What do we notice? What is required to have whatever it is? And let's just say maybe these are enzymes, right? These are enzymes to form a certain structures in the crest. What needs to be present to have a walnut crest? A big R and a big P. What needs to be present to have a P crest? Just a big P, right? What? And how about with the rows? And how about with the single or the foghorn? Recessive. Here's our 1, 3, 3, 9 ratio. And so here the only difference is that you're just looking at one single character. But what is that telling us? That's telling us that the genotype of at a particular gene is influenced by the genotype at that other gene. Okay? And this just spells that all out. It's a single trait with two genes. Okay, so single trait with two genes, we went from having with two genes, and if both genes are influenced in a single trait, we have 4 different phenotypes of that trait, right? What do you think would happen if we expanded that to, rather than having 2 genes, having 3 genes? How many different phenotypes would you have? So if you have just one gene, you have the 2 phenotypes, right? If you have the 2 genes, you have how many? 4 phenotypes. If you have 3 genes, you'd then have 8. Remember that the number of squares expands exponentially or non-linearly, but the number of phenotypes is just linear, so it'd be 8. How about with 4? 16. How about with 5? 16 times 2? 32. How about with 6 then? 64. So here we look at human eye color. Everybody always, Whenever I tell them I teach genetics, they always say, "What's dominant? Blue eyes or brown eyes?" And what's the answer? Something that's really going to bore them, right? Because there's going to be a whole bunch of different colors of eyes, right? And we see that. That's why we have this continuum. So the more genes that are influencing a trait, the more categories. How many categories did we decide? 64, and that's in the simplest scenario. There would be 64 different categories if there were only 2 alleles in each of those genes, and those alleles were interacting in a dominant recessive fashion.
Do we know how many genes are for skin color? Because that's a
Um,
wider variety.
I don't know off the top of my head, but it's going to be something similar to that. You're going to see a huge variation in that. And not only that, this is like a minimum. There's really a minimum of 64 different eye colors, but really there's probably more because there's probably more alleles at each of those genes, and there's probably more complex interactions at each of those. And so that's why. So do we have a general, can we describe it in a general sense? Yeah. If you have, a suite of these genes and you're passing those on to your offspring, and both of you have this suite of genes that give you kind of light-colored, bluish, green eyes or whatever, there's a chance that you're going to have a high incidence of that. But then every time, and that's why it's almost like a trap when people ask you that, because as soon as you say, if you were to decide to say, oh, well, I don't want to go into this 6-gene explanation, and I don't really want to draw them a Punnett square, and you say, yeah, dark eyes are dominant, right? So you decide you just don't want to get into it. Then what do they always say? Oh, well, I know such and such, and they both have dark eyes, and they had a green-eyed kid or something like that. And why is that? Because there's these different combinations that are going in. I have a little sister who married a guy from Guatemala, dark skin, kind of has an afro, dark hair, and all their kids have red hair. And so again, why is that? It's the what's that? Mailman, is that what you said? No, it's because there's all these combinations of genes that are coming in. What's that? I know, exactly. Okay, I'll tell my sister you said that. All right, so it gets more and more complex and as you get more and more genes, or you get more and more phenotypes, and say you were to chart those phenotypes, right? So instead of just having one or the other, right, we're now, remember when we did that? We're now talking about eye color where we have just a whole bunch of the 64, right? That's where we start to go from sort of a discrete description of a phenotype to this continual description, because we get smaller and smaller units that are usually less perceivable by us. And so that's why we get, especially in a lot of human characteristics, these continuums, because we have a lot of genes that are influenced. Okay, so genes are influencing each other. There is a particular or a specific kind of influence of one gene over the other gene that we term epistasis. So this is a term that you'll need to know. Epistasis is when one gene is influencing the genotype of one gene is influencing the genotype of another gene, but in a very specific way. It's actually overriding or sort of trumping the gene expression at the other gene. Okay? So Christian?
So if it overrides the other genes, could we really say that it's really the one that controls everything?
Yeah, so you can see, okay, it has more control, but it's like, it just depends on the particular situation. Because sometimes what we'll see here in a second is sometimes it could be the recessive allele at either gene that is overriding. And when we say overriding, we just basically say if there's a recessive alleles, homozygous recessive of one of these genes, it doesn't matter what's at the other gene, that determines the phenotype. So epistasis, whenever you see epistasis, just think overriding. It's overriding the genotype of the other gene, okay? And so the best way to kind of explain this and to help you kind of get a grasp of it is to go through examples. So So let's go back to that cinnabar mutation. If you remember the cinnabar mutation, last time when we were doing those crosses, if you remember, the cinnabar mutation produces flies with bright red eyes. The white mutation produces flies with white eyes. We know a little bit about that because of lab, right? What would happen then if you made a white-eyed individual with, let's just for simplicity's sake say they're true breeding, with a cinnabar mutation? What kind of phenotype will result? When we do that, and we've done that, what happens is the offspring have white eyes. So in this sense, we have one gene that is, coding for that cinnabar mutation, right? One gene where the allele has to do with the white eyes. When you get them both together, the white-eyed mutation seems to override the cinnabar mutation. So it's epistatic over the cinnabar mutation.
Do we know what causes that? Why are some genes epistatic over others?
Why? Well, yeah, that's a good question, Carl. So why is that? Do we know why that happens? In some cases we do, and we can even think about it in this case. So in this case, what's happening? We know that, remember when we were looking at cinnabar and scarlet? The cinnabar was produced, it was the gene that was producing the red, and the scarlet was the one that was producing which one? The brown, and they both had to be produced, both those pigments had to be produced, right? In order to have the wild type eye. With the white eyes, what's a possibility? That neither of those are being produced, right? But in this case, when we cross them, it says, well, if it's going to be heterozygous, right? So it's going to have the cinnabar, so you would think it at least is producing the red, right? Unless possibly the wide-eyed mutation is something entirely different. That's actually what we see here. And so here, let's just say there's two different genes. We have the cinnabar, we have the wide-eyed, right? We made them, and these are the genotypes and the phenotypes that result from, we'll just do a dihybrid cross with them. These are the genotypes that result. Big C, whatever, big W, but whatever, they're wild type. Big C, whatever, little w, right? Homozygous recessive, that is white eyes. Before we even looked at this, we had a good idea that it probably wasn't the same gene, right? Because we saw in lab that white-eyed mutation corresponded with what? With what kind of mutation? Sex linked, right? So that probably indicated, okay, that's probably on one of the chromosomes, right? And so that's likely not the same one. So if we have little c, and then big W, whatever, we get the cinnabar, right? So little c, cinnabar, little w, wide-eyed. If we're heterozygous in each of those, it's wild type. And so this is the one that's going to determine which one is epistatic over the other one. When we have little c, little w, it's white eyes. Here's where epistasis is happening. The little w is epistatic over the little c. Okay, does everybody see that? Because little c up here is bright, little w is white. If this resultant homozygous individual of both of those genes was bright-eyed, then we would say no, the cinnabar mutation is epistatic over the white-eyed mutation. And so, where is the epistasis being manifested? Here. What is a possible mechanism for it? One, we already know that the bright-eyed is the pigment, right? It's a mutation in the production of the pigment. What we now know, white-eyed has to do with from a physiological perspective, has to do with transportation of that pigment. So you can produce pigment, but it's not going to distribute it within the eye in a way that you can express that eye pigment. Okay, so regardless of what pigment's being synthesized, if you don't have that transport protein to transport the pigment, then you're going to have white eyes. And so that's why it's overriding anything else. You could be producing all the pigment you want in the world, but it's not going to work unless you have that transport protein. Okay, so we can kind of think of it on a physiological level too. So physiological level, not only a physiological level, but we can see it manifest. And for some things, we may be able to look at the genotypes and not, that's the first step after figuring out which genotypes are epistatic, or which genes are epistatic, or which alleles at whichever gene is epistatic over the other ones. That then leads us to our next step. Okay, we need to figure out physiologically what could possibly be going on. So with genetics, I mean, just like what we were doing before, it's always these small steps, and each small step is opening new questions that you then eventually are trying to describe the whole system, and eventually get to a situation or a point where you're describing what you're being able to describe, and then eventually, with transgenic stuff, control that physiological expressions and things like that. So that's why genetics is such a powerful, discipline, and that's why it's spreading to all these other disciplines too. Question, Josh?
On the previous slide,
Hold on, let me pause this real quick so that I don't, and then maybe that'll make some more sense. Okay. Okay, so another example, again, Bateson and Punnett, we're just, going gangbusters on their different, trying to follow up Mendel's stuff. They were looking at sweet peas, which is a different plant than Mendel's, and they noticed that sweet peas the flower colors came in two different colors, white and purple. The purple was caused by the production of anthocyanin. The white flower was the result of no anthocyanin being produced. Okay, but you could have, and they knew you could have, different lineages of white flowers that were parental lineages, true breeding, right? And so they had, this number one lineage, number two lineage, and when you mated an individual from the one white lineage with the other white lineage, you got purple flowers out of it. So there were two different genes influencing the production of this anthocyanin. So what do? When you're trying to figure these things out, first you take these true breeding lines, you cross them, you get the F1. What do you do with the F1? You mate them. They mated them, right? And the F2 generation came out. They counted up the offspring. They tried to figure out a ratio. They did a chi-square test, maybe not, but we would. And the ratio ended up being a 9-to-7 ratio. So now they're thinking, okay, what do you do here? What would you do here? 9-to-3 ratio. Is there anything we can do with that? Can we explain that somehow? Is there anything familiar about that, Carl?
it's almost a 1-to-1.
Almost a 1-to-1 ratio. So you maybe want to keep doing a bunch more experiments, right? And so let's say eventually you said, no, we're going to reject this. We did enough that it's not a 1-to-1. It actually is this 9-to-7. Is there anything familiar about that? Kevin?
It's the 3-to-3-to-1 together.
Yeah, so what do they, but even before that, what do they add up to? What is that ratio? 16. So then you're going to say, oh, wow, there's that 16. Instantly in your mind, it's going to flash that 16 cell Punnett squared. Then what else are you going to notice about it? The 7 is really a combination of which ratios?
3-3-1.
3-to-3-to-1. Okay, we're talking about a single phenotype now. Remember back to the chickens. Oh, okay, we can have two genes that are affecting a single phenotype. In this case, that's probably what's going on. And you're like, all right, cool. But now you have to figure out, okay, what exactly is going on? Why do we have 9 purples and 7 whites? Let's think about this in the context of what we're talking about with epistasis. Okay, so could we write out the genotypes of each of those white lineages? Yeah, we could do it with whatever. Maybe I'll just go to the second slide, the next slide, because I don't remember what letters I used, but say, okay, let's do it. We draw it out, so we ended up with CCPP, right? And so here, we just draw them out. The first one was big C, little p, little p. Second one was big P, little c, big P, right? Know what I have up there? All right, what do they produce? What was the F1 genotype then? This is going to help us just recreating these genotypes. You get big C, big P, little p, right? That's your F1 genotype. Does everybody see that? You then mate that. We know our classic dihybrid cross. We mate that. We draw out our Punnett square. We then plot the purples and the whites. We don't do that. This is just basically what the results are, right? We see that in fact, what is a characteristic then of the purple flowers? What do they have? What is required for the flowers? Or what is required for purple flowers? Or we can even say it a different way. What is required for the production of anthocyanin? Christian, what do you say? Yeah, we need a big C and a big P. Anywhere there's a big C or a big P, then we're going to have a purple flower, right? What is required for a white flower?
It needs to be homozygous in one of them.
It has to be homozygous, what? Recessive
in one, yeah.
in at least one of them, okay? And so now I think, all right, we're understanding this. We're getting closer. Okay, now the next step, though, what do you want to do the next step? You want to try to figure it out on what kind of level? Propose some sort of mechanism on a physiological level. So, could you do that? Could you propose what's going on here based on like gene products? Or maybe for this first one, we'll just go through. When you get to these, you say, okay, something's going on, right? The homozygous recessive at either of those genes is being epistatic over what? The dominant at the other one. So here's a case that's a little bit more complex epistasis. So here, little p, doesn't matter, right? What's over here, because it's going to be white. What happens when you have a little c? Same thing, little c. This one is dominant. It doesn't matter. It's going to be white. And so what you want to do is you want to try to think, okay, let's think of in terms of like physical products or physiological products. We say, okay, we have, at the beginning, maybe we have a white or some sort of colorless substance that we then want to change into eventually being a purple or anthocyanin, right? And in here we have some sort of intermediate one. Okay? So we could then say, okay, what is required to convert this white substance into the intermediate substance? Then what is required to turn the intermediate substance into the purple anthocyanin? What is? Doesn't really matter. We could just say, okay, and you'll do a bunch of these, so you get used to it. So I'll just go through and do it. We'll just say, okay, on this one we're going to say that a product of that dominant big C is required in order to turn the white substance into an intermediate substance. This intermediate substance still isn't colored, right? It's still white. But here, so everywhere there's a big C, right, we at least have that intermediate substance that is still white. So here's a big C, intermediate, big C, intermediate, big C, no big C here, no big C here, big C here. So now we have a distinction between the ones that become purple and the ones that become white. So what do we need to turn that intermediate substance into purple? We need a big P. Okay, so now we're just proposing, we don't know if it's true or not yet, right? We probably have to do some follow-up experiments, but we're proposing that this is what's required to turn the white substance into the intermediate substance. Big P is what is required to turn the intermediate substance into the purple. Here, we can then say, okay, purple, which substance is produced? Purple. Why? Has a big P and a big C. Okay? Does that make a little bit of sense? Hopefully. Here, precursor, intermediate, anthocyanin. This is all just basically what we just did, drawn out in a table form. Okay? Big C, you get the precursor. Here, big C, big P, you get the intermediate. Big C, little p, you get the intermediate, but on this one, that intermediate is not converted into anthocyanin because it doesn't have a big P. So, in this one, the recessive alleles at either gene are epistatic over the genes that again, it's enzymes. Enzymes in the conversion of this pigment are required to finally get that purple substance. Okay, so we're going to go through a couple more fast, and then we're going to do a problem. Okay, here's another example of epistasis. It's in the shepherd's purse. The seed capsule shape in shepherd's purse can be either triangular or ovid, right? If you have a true breeding triangular lineage, take some and mate them with the ovids. In the T1s, you get all triangular. Okay, so again, we have kind of a result that we don't know at that point. We don't really know how many genes are involved. We don't know any of the relationships, but then we mate them like we always do with the F1s. And the F2s, the ratios are 15 to 1 of triangular to ovid. Again, it equals that 16. As soon as we see that 16, what do we tell ourselves? We're like, jackpot, we're familiar, we're comfortable with 16. Probably means that there's 2 genes involved, dominant, recessive, etc. But it's just the one phenotype. If there's just the one phenotype, that tells us that those genes are interacting with each other, right? The next step then is to come up with some sort of explanation for the 15 triangular to the 1 ovid. What would it be?
At least one dominant in order to show the
Yeah, so good, Elizabeth. So we have the 1. We just in our minds we picture that 16 cell Punnett square, and we always know that 1. There's that 1 down there in the bottom right-hand corner. What was that 1 that always is kind of different? It's the homozygous recessive at both of those. You say, oh, good. All right, that makes sense. In our ovid 1, right? It's a homozygous recessive. Here, this is all drawn out. You can go through, we'll just go, we don't need to go through all the steps again, but here's that 1 in the bottom right-hand corner. Little a, little b gives us the ovid. All of the rest of them are triangular. Could you draw this biochemical pathway that you would produce that? Well, and on these, it's not really that important that you come up with the quote-unquote right one, just that you come up with a biological pathway that works, right? So in this one, what do we have? We'll just call it triangular or ovid. And so in this one, well, I won't even draw it up. I'll just go to the next one because we're almost out of time. The pathway then be this. You have a precursor, and then you have a product. And each of those genes, either of those genes, right? Whereas before we needed both of those genes to get the complete product, right? In this one, you can either have the big A or the big B to get the product that equals that triangular phenotype. Okay? And so this is just a proposed biochemical pathway in that production. And then you can go through and you can write out the genotypes of each of those. Triangular, triangular, triangular. They have to have at least one dominant allele at either of those genes. That's where the epistasis is happening. Okay? Epistasis here. The big B is epistatic over the little a, little a. The big A is epistatic over the little b, little b. Either one. Okay. We're not getting to the. Well, okay. So we'll hurry on. Yeah, we'll hurry through this one because I want to give you the problem that you can do between now and Tuesday. So here, squash. How many of you raised squash? I raised squash too. We have white, we have yellow, we have green, different kinds of squash. I don't know exactly which variety that we're talking about here. Same thing happens. White squash, we mate them together. We get a white squash out of it. We mate the white squash together, right? These F1 whites together. We'll just call this the F1 just to be consistent. We'll just call that the F2. Here we have our 16 cell. Squares. Yeah, maybe we should just go. Try to figure out this one. We won't do the problem. All right. So we have a ratio of 12, 3 to 1. Try to figure this one out. Then, if you want, you can look to the next slide before next time. And then when we come to class on Tuesday, we'll actually do a problem where we're not going to show you any of this. That'll maybe be one you'll write on a piece of paper and hand in. try to figure out this ratio, 12:3 to 1. Again, it's 16, right? So we know it's 2 genes. Okay, see you next time. Okay, so let's at least set it up, right? What do we always want to do in these story problems? You want to figure it out, okay, what's right, what's going on. And so you look at it, it says Drosophila wild type are red, you know, little w are white, right? Little w white. Little y, yellow, right? So you have mutant strain, mating a white-eyed fly with a yellow-eyed fly gives you wild type, right? So white-eyed with yellow-eyed. These are two different genes, though, right? So you could say, okay, this is our white-eyed, little w, big Y. That's our white eye, right? What then would be the yellow eye? Big W, little y. That's our yellow. And that, mating those two together, would give you what? Big W, big Y, little y, and that's your wild type. Okay, so we'll stop. But now I'll let you move on a little bit, okay? So did that help? Any of you get started? A little bit more? Okay, now. Okay, so the next thing that you do, okay, so you know the cross that's going on, right? So here, we're familiar with these kind of crosses, right? Dihybrid crosses. So this is something we've done a whole bunch of times. You should automatically, it should almost be like an instinctual just reaction to it, right? Knee-jerk reaction. Oh, this is a dihybrid cross. We start thinking about that Punnett square in our head, a 16-cell Punnett square, et cetera, et cetera. You then go to the results. And I like to write the results down in a column. And so you go red, 92, yellow, 32, white, 41, add them all up together. There's a total, is that right? 165. What do we notice about this? If I just were to circle this, 9, 3, 4, 16. That reminds us of our 2-gene system, 2 alleles, blah, 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 right? 9, 3, 3, 1. What's the only difference? 9, 3, this 4 is the 3, 1 part of the ratio just added together, right? So here, 9, 3, 3, 1, 9, 4, 3, 16. What would we then do if we were doing it as like a research project? What would we then do? What would we would say, oh, this system seems to be a combination, you know, or an iteration, or not an iteration, but yeah, sort of a combination of that 9, 3, 3, 1. It's a 9, 3, 4 ratio. But to be certain, what do we have to do? This little thing, what's this little thing? Chi-square test. How would you do that? This is what we'll do with the corn today. You would do that by taking n and timesing this one by what? Here, these are your observed, right? Could you get an expected from that? You just do 165 times what? 9/16, right? So you do a 9/16, oops, sorry. 9/16, 3/16, and a 4/16. You get your expected. You can then go through and do a chi-square test with that. Okay, what would your degrees of freedom be? 3. How many phenotypic classes are there? Yeah, so here's where there's a change in the degree of freedom. There's only 3 different phenotypic classes. So you have degrees of freedom too. Okay, but you don't need to do that right now. But, you know, on the test you could. It's something that you could do. So now we say we got our ratio, right? Now we have to do the last part, which is kind of the sort of the most new part for you to do, and that is to figure out the biochemical pathway. All right, now we're all up to the last part, so let's try to figure out that biochemical pathway. So the next step you want to do is you want to figure out which genotypes are giving you each of these. Which genotypes are giving you the 9? Which genotypes are giving you the 3? And which genotypes are giving you the 1? That's your next step. So how many people have done that step already? Figuring out the genotypes for each of these phenotypes and ratios, right? The 9 wild types, the 3 yellow, and the 4 white. How many of you figured out which genotypes belong there? Okay, so there's a few of you. Okay, let's take a minute and figure that one out. Yeah, so this is actually, how many of you have done this sort of thing in your genetics classes or your biology classes before? No, so that doesn't surprise me. This is usually the kind of stuff that is brand new to people. Whereas how many of you have done dihybrid crosses, even trihybrid crosses? You've probably done that stuff before a little bit. We're kind of tweaking it. But this, I know that this stuff is probably the first time for a lot of you. That's why we're kind of going a little bit slower. So bear with me. Oh yeah, I said I would. Yeah, hand it in to this side and I'll get it. You want to take it? All right, just keep it. Yeah, because I forget, we went through it anyway together, and so you're all going to be pretty much have it. So yeah, so keep it. Hand it in a lab today. How's that? So you can go, you can look it over, and then hand it in a lab. Okay, so another phenomenon that we're going to talk about is pleiotropy. Pleiotropy is something that we all probably, especially if you've had biochemistry or whatever, it's something that's very intuitive to us. I pretty much guarantee you all kind of already understand it. Basically what it is that if your gene has some, a mutation at a particular gene can have a cascading biological effect on a whole bunch of other aspects of a phenotype. That's all that it's saying. And so the two examples that it gives in the book is with phenylketonuria. So you have a mutation in this gene, right? You're not able to make that enzyme, right? You know, going back, it ends up causing a whole bunch of different things. We saw that. If we go back to another example, albinism, right? Mutation in that gene causes a whole bunch of effects. What are some of the effects of somebody who's phenotypically albino? What are some characteristics? White. What else? What color are their eyes?
Red.
Yeah, so they have the irises are kind of this pinkish color, right? They're sensitive to the sun. There's a whole bunch of things that result from that. We're used to that. We're used to that because we look at on a more chromosomal level, and we'll talk about it in the next chapter, Down syndrome. If somebody has an extra chromosome, trisomy 21, there's a whole bunch of effects, right? What are some of the effects, the common phenotypic results of having an extra number 21 chromosome? Shortened stature, right? What else? Facial features. There's usually morphology of the tongue, hand, the lines in your hand. There's a whole bunch of different ones. We're used to that because that's in a lot of syndromes and things. There's a whole suite of things that happen. That's all we're talking about with pleiotropy. So, for instance, with phenylketonuria, there's mental impairment, light hair color, and the presence of metabolites in the blood and urine. That's something we can test for, right? Mutation in Drosophila, the SING gene, affects the bristle shape and the egg production. So it's not just affecting one thing, it's affecting a whole bunch of things. That's all pleiotropy is saying. So here, the next slide is just an example of that phenylketonuria. Here, this is just the chemical sort of pathways. If you look up at it here, by not being able to convert phenylalanine, you get a buildup of different things and blockages of different things, and it has this cascading effect. Don't worry, you don't have to remember this at all. I'm just showing you this to show you that it can have this cascading effect. Okay, that's it. Pleiotropy is compared to epistasis, pleiotropy is, a cinch. Don't worry about it. Okay, so that's all. All right, so now let's move on to the last part. The last part we're going to, again, like we did in chapter 3, we're going to talk about human stuff. Okay, so here you can go back, you can read points on your own. The aspect of human genetics that we're going to talk about in the last part of this chapter is inbreeding. To talk about inbreeding, to look at inbreeding, we're going to go back and we are going to look again at pedigrees. This time, how many of you feel fairly confident? You understand pedigrees? You can read a pedigree pretty good? Well, now we're going to just tweak them just a little bit. So we have to tweak the pedigrees a little bit to look at inbreeding. We do that because we want to be able to calculate a metric. this metric that we calculate for inbreeding is called the inbreeding coefficient, and it is symbolized with a capital F. There's a suite of these statistics that we use in population genetics. They're called F statistics. We may get to them later on in the class. We'll touch population genetics here and there. We've already kind of done some examples of population genetics. Today we'll talk about some population genetics. If we have time at the end of the course, we'll do a whole section on population genetics, and we will look at F statistics. F statistics, if you remember in chapter 1 when I was explaining to you the research that I'd done in Sao Tome and Principe in Africa, what we did with that when we were looking at estimating gene flow. So we do this a lot in wildlife biology and things like that. We do this a lot in even human medical epidemiology and things like that. To estimate that, we calculate F statistics. We can calculate gene flow by calculating F statistics. There's an F of IA, F of ST, F of IT, etc., etc. It's a little mathy, but here we're just going to introduce you to this. The reason I'm talking about this is the inbreeding coefficient is part of that suite of F statistics. Okay, so basically we calculate the inbreeding coefficient because we want to analyze the effects that mating has, or the effects of what we call consanguineous or matings between relatives, inbreeding in a general term. Okay, so to do that, calculate the F statistic, we first look at a pedigree, right? So sort of the first step is always looking at this traditional pedigree. This is just exactly the same as your other pedigree, right, that you're looking at. We have one new kind of designation. You see this triangle designation. What do you think that means? Twins? Yeah, no big deal. The other, what's another something different is the double bar. The double bar just means consanguineous matings. Consanguineous means what? Sanguineous means what? What's that? Blood. Yeah, blood. Sangre, right? Sangre with blood, right? Yeah. So consanguineous means with blood. Okay, so here, although we see here that we have this mating between relatives, with the mating within blood or with blood, right? I think that's a typo. Unless the only way this couldn't be a typo is if it's saying, okay, this one we know from outside information that they are actually relatives, but we don't show this pedigree. But I think it's just a typo, so kind of ignore that one, okay? And so here we have an actual pedigree, right? This is within an Amish community. What do we see is this trait. We can go back to like when we first learned about pedigrees. We can say, okay, what's our first question? We're going to ask, is this a trait that is what or what? Is it dominant or recessive allele? In this one, what would you say? Recessive. Why? Because you're getting it oftentimes, and in this case, always, right, when you're getting these matings between relatives, consanguineous matings. Question?
What is the three-circle?
Yeah, that's another good one. So this we will see this one again. This three-circled means that for spacing issues, we're just going to say there's 3 females in this rather than bringing it all the way out here. This, on the end, is not as big a deal, but a lot of times when they're doing these pedigrees, there's spacing issues, right? So like if it was right down in one of these or something, you might see that. So that's a good question.
So with the one in the middle with the flower kits, they could have done a circle with the 4 in it?
They would have wanted to do 2 circles because they would have wanted to have a circle of somebody who's affected and not affected. But they could have had, but then they have this twinner, so it's a little bit weird. So this one could have been a circle with 2 in it, a square, open square with 3, and then a red square. So yeah, you can do combination of that. Then that's just a graphical aesthetics. Well, let's see, who is first? All right, Pat.
I was going to ask, so albino only come from inbreeding?
This is a good question because this is exactly what we're getting at. Does albino, what does inbreeding do? That's the overriding question. Yeah, so what do you mean by recessivity? All right, hold on to that answer. That'll change a little bit. That's what we want to get at, right? That's what we want to get at. We want to get at what exactly does inbreeding do, right? So here we have an Amish community, maybe, I don't know where it's from, Birmingham, in Pennsylvania, or somewhere where there's, you know, high community of Amish. We see this in oftentimes communities that we would view as maybe closed for other reasons. We have, you know, there is some, just by necessity, right? Here, these are pretty, you know, these are high levels, not high, but kind of high levels of inbreeding, right? But in effect, there's probably low levels of inbreeding that are going on all the time, right? Simply because of historical facts. When the Mormon pioneers first came to Utah, within a couple of generations, there's probably going to be some inbreeding going on, right? When, you know, the beginning of the 13 colonies in the Americas, same thing. Why? Because we oftentimes find populations with low numbers of individuals. We see cases where different islands have been started by, you know, 24 people. Again, obviously not so much now because we were able to travel a lot more, right? But 100 years ago, they're going to be confined to who they can mate with. So low levels of inbreeding are going on all the time because of historical reasons, or even because of cultural reasons. So here, for instance, the Amish, you know, are coming culturally. It's a religion, and so they usually marry within the religion. Do we have examples of that even today here in Utah, or across the border in Arizona? Just across the border. How many of you have been to Colorado City? Anybody? So you have been to it? Okay, so Colorado City is the FLDS, right? And so the fundamentalist LDS church, Warren Jeffs is in the news and everything. So in that kind of community, you're going to have higher levels of inbreeding going on just because mating is going on within this closed community. Okay, it happens lots and lots of different places, Amish, wherever. So what is the effect of that? Do we see it? Have we seen it in other, maybe larger scale cultures? Yeah. But historically, we've also had and known just intuitively that oftentimes these consanguineous matings oftentimes produce children that are, you know, debilitated or diseased, or higher incidence of that, right? And so we've passed in a lot of cultures different, you know, laws or whatever, either between religious leaders or civic leaders that have to give the okay for, you know, first cousin matings or whatever. But why is this? This is what we're getting at. Why does that happen, Elizabeth?
Because you have such close genetics, there's an increased likelihood of having the debilitating genes.
Okay, so there's an increased likelihood of having them. Did you ever hear that? Why? So why is that? And can we quantify it? So why is, we will be able to see why. We can see why in the pedigree. But what the inbreeding coefficient does is we're going to start quantifying that. And so that'll be important, and again, it's going to deal with probability. Okay, so why is that? If we look around other communities that have high levels of inbreeding in ancient Egypt, especially with, you know, the pharaohs and the lines of the pharaohs, there was lots of inbreeding going on. In different royal lineages in Polynesia, there are also a lot of inbreeding. In India with the caste system, not as much anymore, but historically in the caste system with the Brahmas, if you were a Brahma, you don't marry an untouchable, right? And so again, increased inbreeding in Europe. Where in Europe does a lot of inbreeding go on, or did go on? Maybe still goes on a little bit.
English ruled the royalty.
Yeah, anybody watch Downton Abbey? So does that happen in Downton Abbey?
No.
Right? Who did Mary marry? Cowleys, right? And the Granthams that are married, or distant cousins, they got married, right? So it happens. Nobody else watches Downton Abbey? I like that show. All right, so that happens. Royalty. The good thing about the case with Europe is that for a long time they were really keen on keeping good records, and so we have good pedigrees, and then we also have a fairly good knowledge of individuals that were. Okay, so is there a biological mechanism then, if in fact, and you can think about these in evolutionary terms, if in fact there is a penalty for mating with somebody who is closely related with you, and we're going to quantify that here in a second, is there then, or should there be a biological mechanism that helps you to avoid that? Raise your hand if you know.
In that video?
All right, that's what I'm getting at. All right, raise your hand if you saw this science of sex appeal. In that video, what was the mechanism? Yeah, the smell that was giving a hint to the MHC alleles. And so, yeah, so here, back, yes, that video was fun, but yeah, we learned something, right? And it plays, it comes into play in part of the lecture. Okay, so yes, there should be a biological mechanism. We didn't really know it, but now we're kind of beginning to describe that biological mechanism. Okay, so in general, we call this effect, this sort of the sum total of this effect, especially when we're talking about food crops and stuff, we call that inbreeding depression. Here, this is an example of lines of inbred corn. This is one line of inbred corn. This is another line of inbred corn. These lines of inbred corn, we could also call those true breeding parental types, right? Remember, we've been talking about all semester. You see the size of this inbreeding 1, inbreeding 2 line, small. You breed this line with this line, you get the hybrid. And what does the hybrid look like? Much more robust, much bigger, etc., etc. We call this effect of the breeding of 2 inbred lines and having a hybrid that is more robust. We call that hybrid vigor or heterosis. Those are just 2 terms that we call. Here, these are ears of corn from the same lineages. And so we see that there is an effect on if we have highly inbred lines. We know that. How many of you, are there any pre-vet people in here? Pre-vet? People are going to be vets? Okay. Is there anybody with a dog in here? Okay, so what do we know about lots of different varieties of dogs? How many of you, I've had a couple of yellow labs. What do we know about yellow labs and golden retrievers? These varieties are oftentimes highly inbred, and what happens to them? They have hip dysplasias. They have hip problems. Okay, we see that with lots. With is it basset hounds or beagles? These are highly inbred. One of the issues with beagles is what? They have spinal problems, right? And so we see that. We know that from just our own experience with our pets and with our agricultural products. Okay, so it's something that we want to avoid. All right, now let's go into what is the real effect. Like, what is it? What's really happening? So, inbreeding, we know in general, inbreeding increases homozygosity. So when we're looking at that pedigree, especially when we're talking about recessive characters, right, or diseases or whatever, like go back to albinism, diseases or conditions that are caused by homozygous recessive, if inbreeding increases homozygosity, just in general, we're going to see an increase in 2 recessive alleles getting together. Okay, there are lots of rare alleles in populations, and when we talk about population genetics, we're going to actually calculate, and we're going to look at 3 populations in Hardy-Weinberg equilibrium, and we're going to say, okay, why are there so many rare alleles in the population? I've read somewhere that we all have, and I think I mentioned this before, at least a dozen recessive deleterious alleles in our genome. They're kind of hiding out there as heterozygous, in the heterozygous condition, and we'll look at that a little later. How can we quantify this increase in homozygosity? That's what this inbreeding coefficient is all about. Okay, so what we're going to do. All right, genetic analysis of inbreeding. So we have a couple of minutes. Let's just do this slide. It'll get you ready for this new pedigree. Okay, so here we have our pedigree. Now, rather than having in our old pedigree, we would have this circle, which is the female, and the male mating, right? We would have them mating, and then we'd have a line that would come down to the offspring here, the box A. So D and C mate and they produce A. So rather than having it that way, the only change in this pedigree is that we slant the lines down. So A is a product of a mating between the male C and the female D. B is a product of mating between the female E and the male C. Okay, I is a product of mating between the male A and the female B. We're still labeling these generations and things like that. So that's the only change in the pedigree. The reason we change this pedigree, or how we're doing this pedigree, is that we want to identify these things here in the middle, and these are called inbreeding loops. So in order to calculate this inbreeding coefficient, we have to identify these inbreeding loops. Okay, so here, within each of these individuals, these are alleles. So we're just going to think of them as these little particles. And so what we want to do is we want to calculate the probability, and so we're just looking at one gene, right? We're not even going to, maybe these are separate kind of alleles, maybe not. We're just, the little dots in here are just any allele, right? They each have two alleles with C, two alleles with E. We want to calculate the probability that one allele, one of these two alleles in C, can get passed down to I is the inbred individual, through and come together as a homozygous condition because it has been passed down that loop. So here's an allele. We want to figure out probability that this allele gets passed to here, then to here, right? So it passed from C to B, from B to I. We want to see that allele, probability of that allele also being passed from here to here and here to here. So again, we're going to be talking about events. We're going to talk about probability of events, etc. When two alleles come together in I and are the same, homozygous recessive alleles down here are the same, because they got passed down in that fashion, we term that they are identical by descent. Okay? And so here, the important thing that we're going to do is we're going to, in our pedigree, identify the inbred individual, identify the common ancestor. From every common ancestor, we're going to look at a loop. We're then going to calculate the probability that these alleles down here are identical by descent, add all of the loops up together, and that's going to give us our inbreeding coefficient F.
So in the box of the C, those would represent two alleles, like a capital C, small c, lowercase c.
Yeah, or little c, little c. It doesn't. The reason it's just two dots right here, it doesn't really matter. Later on, in a few slides, we'll actually give you a blue one and a green one, and so that we can track the pathway of
So you
each of
just
those.
use one half chance of them getting one of those little.
Yeah, so these are events one half. We'll get to that later. There is an easier way of doing it. There's a formula that we'll do, but we'll wait. We'll do that next time because we're at this. We're out of time. We're done. All right. So that's what we'll do on Thursday.
So the dots are
Okay, here you are. Calculate the inbreeding coefficient for half-sibling matings, all right? So this, you could, looking at this pedigree, see, okay, here are the two individuals that are doing the matings. They share a common ancestor. That common ancestor is what? The father, right? So they're half-siblings. They get together and have an individual here. You identify the inbred individual. They make it easy because they call it I, right? I for inbred. They make the common ancestor easy. They call it C. That's for the common ancestor. Okay, so here you identify, can everybody see the loop? Usually when we define loops, we just list the letters. So this loop would be I, A, C, B, the number of individuals, and I think this is your question, right, Christian?
Yeah.
The number of individuals here are A, C, and B are the number of individuals in that loop. So you take 1/2, raise it to n, which equals 3, the number of individuals in the loop. That equals what? 1/8. Okay, so the inbreeding coefficient for half-sibling matings is 1/8. And here, this just you through it. This next slide. Carl, question? Okay, so the question was, why aren't we counting the inbred individual? And this will make, so hold off with that because we're going to look at the probability, right? So we're gonna look at it in terms of probability, and we'll do that in a second. Okay, so here, this is just taking you through each of these steps. You can't see there's a yellow 1, yellow 2, and yellow 3 here, so maybe those of you in the back can't see that, but you just go through, you count the individuals, you identify the common ancestor, you then raise 1/2 to the 3rd. So if we think about it, so getting back to Carl's question, if we think about it in terms of probability, you're basically again calculating, right? Let's just take this. Let's just look at one allele. We're not going to do the total calculation because we'll do that here in a second. But just looking at one of these alleles, so I'm going to take this, the allele of C on your right-hand side, right? Probability that it gets passed down to B is what? Probability that it gets passed down here, 1/2. Probability that it gets passed down here. Probability that it gets passed down here, 1/2, right? But that's only one of the alleles. And so we'll have to do the same thing for the other one, okay? So the reason you don't count this individual is because you have two different pathways, so then you don't. I mean, there's no like theoretical way. That's just sort of a mathematical way. There's two pathways, so you count one less individual, or you times it by one less, times it by 2, times it by 2 takes out one of the 1/2s. So it just works that way, right? So don't think about this sort of 1/2 raised to n as a just consider it a shortcut, but we'll in a slide or two we'll get to the actual theoretical that'll make sense. We'll do it in terms of events and probabilities and stuff like that, and you'll see that it's the same. Okay, so that's a good question. All right. So here, let's do one that's a little bit more difficult. First off, can everybody see how that is a full sibling mating? So we go to this first generation, right? You go to this first generation, U is the father, right? V is the mother. So this makes the pedigree a little bit more complicated, right? So instead of just having it go to one individual that we have over here, we shift it over here and shift it over here. So S and R have the same father and mother. Did we see that? So here's the father, here's the mother. They're full siblings. They decide, okay, we're going to be together. And so could we take this through the same sort of methodology that we just did and calculate the inbreeding coefficient? You might think just intuitively, okay, we looked at a half sibling, right, mating, and now we're looking at a full sibling mating, so what would we expect the inbreeding coefficient to do? Go up or down? Up, right, because there's a greater probability now because there's two different pathways. So how do we do this one? So let's go to the next slide, and the next slide takes us through it step at a time. Okay, identify the common ancestors. Can we identify the common ancestors? Shout them out. U and V, right? Okay. Oh yeah, I guess you could just read it. Okay. Common ancestor, U and V. Count the number, well, first, before we count the number of individuals in the inbreeding loop, we got to count the number of inbreeding loops, right? So here, Z this time is the inbred individual. Here's one loop, right? Take an allele from U, that one allele can be passed down to R, can be passed down to S. That allele can then be passed down from S to Z and R to Z. There's that one loop. We'll call that loop 1. The second loop is the same thing, but from common ancestor B. Does that make sense? Okay, come down here, comes down here. So we have 2 inbreeding loops. Loop 1, U, R, S are the individuals. N equals 3. Loop 2, V, S, R are the individuals that we counted in loop 2. We calculate those out, 1/2. And now remember, you have to calculate the component of the total inbreeding coefficient for each loop. There's 2 loops here. You count it out, that's 1/8 for loop 1. That's 1/8 for loop 2. Therefore, the total inbreeding coefficient F is 1/8 plus 1/8. That equals 1/4. Okay, so now, how likely, comparing a half sibling to a full sibling, how much more likely is an allele in that inbred individual that results from a full sibling mating as compared to a half sibling mating? How much more likely are you to have it? Twice as likely. Okay, and that sort of, once we start doing more of these, this kind of makes sense. Okay, is everybody good with that? It's a pretty easy calculation. I mean, it's an easy calculation. The difficult part is just figuring out the loops. Okay, so let's go and look at, this is where we were with Carl, look at the theoretical basis of that. Okay, so in this one, we've color-coded the alleles. It's still 2 different alleles in a gene. We have one allele that's red, we have one allele that's blue. We're going to count the probability that this red one in C in case 1 gets passed down to I through what we know from the last chapter about probability. We're then going to calculate the same thing for the blue allele here in case 2. So let me look for a dry erase marker. So in case 1, we are calculating the probability that the red allele gets passed down from C to A. Okay, what is that in terms of mathematical nomenclature? 1/2. Well, that's the calculation, but what do we call it? The outcome of a process, right? What's the process? Passing the allele down. What do we call the outcome of a process? Remember back to, is it last?
The event.
The event, right? So here's an event. Event 1, and so we have, okay, event 1 has to happen. So in order for 2 red alleles to come together down here in I, we have to have event allele being passed from C to A, and then another event, right, allele being passed down from A to I. So we have 1 event, 2 events, the second event, sorry. We then also have to have a third event. C has to pass down that red allele to B, and then B has to pass it down to I. So this is our 3rd event and our 4th event. So now back to what Dan said, what's the probability that the red allele gets passed down from C to A? 1/2, right? And doesn't take rocket scientists to figure out that's the same probability for all 4 of those. Now we have 4 events, right? So in order for the red alleles to be identical by descent down here in individual I, 4 events have to happen. Event 1 and event 2 and event 3 and event 4 have to happen. What do we do when we hear and? Multiply, right? So 1/2 times 1/2 times 1/2, that equals 1/16. Okay, that's the total probability that the red allele will be passed down through that inbreeding loop and come together as a homozygous in a homozygous genotypic state in I. We can do the same thing. I could write it all again on the other side of the board for blue, but what do we know? What is the probability of the blue one being passed down? 1/16 2 also, right? So you can have alleles that are identical by descent in that inbreeding, through that inbreeding loop by doing it as we illustrated here in case 2, or doing it in case 1. What do we do when we're calculating probability and we hear or? We add them together, right? We did the and, we multiplied, we do the ors, we add them together. 1/16 plus 1/16 equals 2/16 or 1/8. That's the total probability. Okay, now go back to how we did it. How many individuals in this loop? 3. 3. Remember, we don't count this individual. 1/2 raised to the third is what? 1/8. That's why it's the shortcut to 1/8. Tyler?
In inbreeding, aren't we only worried about the recessive rare alleles?
No. So we in certain cases, that's the ones that we're more concerned with. We're more concerned with the recessive deleterious alleles. But when we start looking at population genetics, there's another thing that happens with, well, it's another thing, but it's kind of the same thing, is that it causes the population to march towards homozygosity. So we're also losing genetic diversity. And so in that case, we want to figure out, we want to know what's happening to the entire genome of these individuals in these different populations. So that's a good question. So what does this mean? What does this 1/8 mean? It means that it is the, go back to the definition of the inbreeding coefficient. Good, Corey. What is the inbreeding coefficient? The probability that 2 alleles, 2 copies, or 2 versions of a gene are identical by descent. There's another way that we can look at this, and we can define it in another way that also is sort of important. That's saying that 1/8 or 1/8 probability of 2 alleles being identical by descent, right, in that inbred individual. That also, you could also interpret that as 1/8 of that individual's genome is going to be what? Automatically homozygous because of being identical by descent. You also are going to have, without inbreeding, you're going to have a certain percentage of that individual's genome that is homozygous. So that's going to convert then the baseline level of whatever that individual would have been, sort of quote unquote normally, how much that individual, what percentage of the genome would have been homozygous, you're going to add 1/8 to that. So as you get generation after generation of inbreeding, what is that going to do to the individual's genomes in those generations? They're going to become more and more what? Hetero or homo?
Homo.
They're going to be more and more homozygous, right? You're going to lose your genetic diversity. And then going back to the slides that we were looking at the corn, what's that going to do to the overall fitness? It's probably you're going to then suffer from more of that inbreeding depression, right? Okay, so those are 2 different ways of looking at that. Okay, so let's try a couple that are a little more complicated. So take out a piece of paper. We'll have this one, you will hand it. Take out a piece of paper. Here are 2 different scenarios. The first scenario is first cousins matings. The second scenario is uncle-niece matings. Okay, so you're going to calculate the inbreeding coefficient for that. There actually was, so we're going to calculate the probability of that. Well, we'll talk about it after you do the, let's take, let's say let's take 5 minutes maybe and calculate it for both of those. When we come back, we'll talk about semi-recent, I think it was about 10 years ago, maybe a little bit more, that the New York Times did an article on first cousin matings. So if you get done early, try to think of historical examples of first cousin matings, because there's a bunch of them. Who feels confident enough to come up and do the first cousin one? Anyone? All right, Dan. Oh, okay. Dan and Carl come up as a team and do it. No. Has anybody done the uncle? Have you either of you done the uncle niece? All right, Carl do the uncle niece, and Dan will do the first cousins.
I think I got it. I mean, I could be completely wrong.
It's all right. This is good.
You
See where the uncle-niece loops are smaller than the first cousin loops. Did everybody see that?
Yeah.
Uncle-niece loop, first cousin loop, first cousins loops. What does that then mean? How do we interpret that? With an uncle-niece mating, how much of the genome will be now homozygous due to inbreeding? 1/8 of it. That's going to be an increase, right, of whatever the percentage is, right? So like 13, 14, whatever 1/8 in the percentage is of genes that are now going to be homozygous because of that. That's why we have that effect if those individuals have a lot of, or even a few alleles of deleterious recessive, of the deleterious recessive type, that increases the chance that those alleles are going to get together or become homozygous in that inbred individual. First cousins, 1 in 16. Okay, so 1 in 16 isn't, obviously uncle-niece is a more serious situation than the first cousins. First cousins, 1 in 16. That's why the first cousins matings has sort of been throughout history. Sometimes there's been relaxed, adherence to avoidance of first cousin marriages. Sometimes there hasn't been. If we go back, not even 100 years or so, we see that there are 200 years, etc. We see that even outside of those conditions, or outside of those situations that we talked about before, where we're talking about royalty and aristocratic lines and stuff, we saw a lot more, much more frequent first cousin matings. We have examples of first cousin matings of people you probably know, or you've heard of, not necessarily know. Does anybody remember any first cousin matings that you've heard of? Yeah, Jerry Lee Lewis and his, not only was it first cousin, but I think he was like 30 years older than her or something too. She was like 16. That was a pretty scandalous one. Edgar Allan Poe is a cousin. Was it first cousin for Edgar Allan Poe?
And then there was Hitler.
Yeah, Hitler. Hitler was the product of one. Albert Einstein.
Darwin and his wife, like.
And most pertinent for us biology majors, Charles Darwin wasn't the result of a first cousin marriage, but he did marry his first cousin, and he had a kid named Emma who actually suffered from a genetic disorder and died young. That was the biggest event. How many of you have had evolution in here? Did you guys go through the life of Darwin much or not? So what was the big event that caused Darwin to become basically agnostic? Did you talk about that? Because Charles Darwin started out going to, become a country parson, country priest, which was a very common thing for people that were really into, the natural world, into natural history and stuff like that, because you could, cruise around, kind of like A River Runs Through It. Has anybody ever seen the movie A River Runs Through It? Remember the dad, what did he do? He preached on Sunday, and then the rest of the week he fly fished. And that was really common back then, that you would go and look at God's creation, right, during the week, and then make a sermon about it on Sunday. So that's what Darwin started out as, but eventually he became agnostic, which was partially mostly due to his daughter, not Emma. Emma was his wife. I can't remember his daughter's name, but she died a pretty gruesome young death as a child, and that's kind of what caused him to become agnostic. Anyway, so Charles Darwin. Christian, you have another example, or?
I actually have a question on this. I was trying to make sense between the example before this. Can you go back a slide?
Okay, here, let me, to go back, I'm going to have to escape out. Okay, so here are a whole bunch of other scenarios. We have full sibling, half sibling, first cousins, double first cousins, uncle, niece. So we're getting a lot more complicated ones. I gotta pull this all the way down. This shows you what the inbreeding coefficient would be for each of these. So you can kind of use each of these scenarios, go through, try to calculate an inbreeding coefficient, and then, after you're done, check it with your example. Okay, so this can kind of be like homework problems for you. So in this one, we won't go through, but let's just briefly identify the loops here. How many inbreeding loops are here? 2, here, and here. How many inbreeding loops are here? 4. So can everybody see? So here's 1, right? Here's another one. Here's another one. And then here's another one. So when you get into more complicated scenarios, rather than just, you can, for the simpler ones, it's easy. You can just kind of identify those loops. For the more complicated ones, sometimes it helps to start with the inbred individual and then just trace a path down, or start with a common ancestor and then just trace a path down to the inbred individual. So there you trace that one path. Here on this double first cousins with the female on the left, you trace it again down a certain path, okay? So that's what makes it, you know, the more complicated ones, that helps. So let's go to the next example, which is even more complicated, and this is going to be as complicated of a scenario as you will see. So if you can crank this one in like 2 minutes. Oh no, that's not going to be the most complicated. This is a complicated one, but this isn't quite the most complicated one, but it's kind of complicated. So try to do this one. Do this one. What's that? No, hand in that paper and then do this one in your notes so you can have it, because it would be good to have this more complicated example in your notes. So hand in your ones to your left, and I'll come pick it up while you're trying to do this one that's a little bit more complicated. So okay, the 3 loops were easy to find, right? The 4th loop is the one that's a little bit harder to find, and is the one that I can only do it personally if I take the alleles from the top and trace them down. Okay, so we're going to trace for the mystery loop, right? The mystery loop, we're going to trace them down. Okay, so LP mystery, we're going to start here in E, right? So we start there with E, and then we get the allele passed down here to A. There's the allele. And then the same allele getting passed down here to B. Does everybody see that? So we go from E, we'll just draw it out as different. So E, it gets passed down to A, right? And then it gets passed down. Actually, we're going to do it a different way, just to illustrate it. We're going to have 2 arrows. So E gets passed down to A. It's almost going to be like our forked line thing, right? E gets passed down to A, and then gets passed down to B. Okay, now in our loop before, A would pass it down to D, right? But in this one, you can see that A and C are also connected. So A could also pass it down to C, right? And B, instead of passing down to C, it could pass it down to D. So it's kind of flipped. So you have one going this way, and then the other going this way. So rather than coming straight down, they're crossing. Okay? So B then gets passed down to D, right? And then, and here we have, this is kind of a cool way to do it. Now they both pass it down to I. So that was the mystery loop. So there's another loop. So now there's 4 total loops. So the other loop, right, we'll call it loop E. Let's do it again in that same kind of nomenclature. We'll do it our other way. E passes down to A, passes down to B. A, then let's do the traditional one. A goes to D, B goes to C, and then they both pass down to I. So these are 2 separate loops. We'll call it loop E1, loop E2. Okay, does everybody see how those are different inbreeding loops? So graphically, it doesn't quite make as much sense. If you wanted to do it, if you were more comfortable just doing the ones on the exterior, think about it in a different way. What if you drew this same pedigree, but then just switch C with D? You would then go down along the edges again. Okay, so that's the most complicated. So then what would be the total inbreeding coefficient? We had 1/8, right? Plus 1/8. Is that right? Plus, what was the next one? 1/32. Plus 1/32 plus 1/32. This would equal 10/32. Is that right?
Yeah.
10/32. That's your total 16ths if you wanted, or you could do it. I'd accept either, and on a test maybe you might even have a multiple choice, so you might have one or the other. So if you had, I usually don't reduce them that much on multiple choice, because I don't want to add an extra step that you're going to screw up, right? I want you to minimize your screwing upness, right?
You're screwing up.
Okay, so has everybody got that? We good with this one? Everybody sees both loops?
You never have like both of them though, right?
We wouldn't have like 10 30-second and 5/16. Would we?
No, and if I did, you'd have to get
I don't like that.
them right for both of
I
them.
know, you totally.
Yeah, it's a good review of fractions. All right, so let's go to the next. The next complication with inbreeding loops is calculating the inbreeding coefficient in a scenario where the common ancestor is also inbred. And this is a more realistic one, right? Because usually if you go down to a community that maybe has a high incidence of inbreeding, Colorado City or somewhere, you're most likely not just going to find one case where somebody is inbred, but you're going to find generational inbreeding, right? And so you have to take that into account. And so if you think about it here, these are what kind of matings? Full sibling, half sibling? Yeah, we're doing the simple one, half sibling. So basically it's a half sibling mating where the father in that mating, Ca, the common ancestor, was also the result of a half sibling mating. Okay, so how you do this is there's also an easier way, an easy sort of shortcut way to do it. You could go through and calculate all the events, look at all the events, calculate the probability of those events, and then go down from there. We won't draw it out that much. We're just going to go to the easy way of doing it. So how you do that is you add a term to our equation. So rather than 1/2 to the n, here's the 1/2 to the n, we add to that 1 plus the inbreeding coefficient of the common ancestor. Okay? So in this case, both of the inbreeding coefficients, so if you were to calculate the inbreeding coefficient of the common ancestor in that case, we would make the loop, calculate it, it's 1/8, right? Then we now have, that then becomes your f of Ca, that's 1/8. We then times that by the inbreeding coefficient that we would have calculated if the common ancestor was not inbred. But in order to account for that, we have to keep that 1 there, right? So this is our expanded formula. In our initial case, right, when we were just calculating the inbreeding coefficient, we could still use this formula, but if the common ancestor was not inbred, what would f of Ca be? What's the inbreeding coefficient of somebody who, this would be a good test question. What's the inbreeding coefficient of somebody who isn't the result of inbreeding? 0, right? So in this case, it would have been 0. 1/2 to the third times 1 is 1/2 to the third, right? And so this is the expanded formula. So in that case, we go through, we now know, that the inbreeding coefficient for half sibling matings is 1/8. F of Ca is 1/8 or 1/2 to the 2/3. We then go through and calculate this out. 1/8 times 1 plus 1/8. 1 plus 1/8. If we do what's in the parentheses first, that equals what? 9/8. 9/8 times 1/8 times the fractions equals 9/64. So how much has it increased? With regards, what is the inbreeding coefficient of the individual if their common ancestor was not inbred? What was that one? Half sibling matings. 1/8, or we could just to compare it to this, it would be 8/64. So how much has it increased? 1/64. So because the common ancestor is inbred, it increases by 1/64. Okay. So here is another one. Let's see if you can go through and calculate this one. A is the result of half sibling matings, right? So you have a full sibling mating that occurs where the father is the result of a half-sibling mating. Try to go through, let's take a few minutes, go through and calculate this one if you can. Okay, so let's do the F of CA first. What is F of CA? We counted up how many individuals. Here's the inbreeding loop, right? F of CA then equals 1/2 raised to the what? To the third, right? So that equals 1/8. Everybody good with that one? Okay, so now what is the formula for F of I? It's inbreeding coefficient, we'll just call it F
Plus
times 1 plus F of C. What is F of C? Or F of CA, sorry. 1/8. So we have that. We have what's in the parentheses calculated. Now we just have to figure the inbreeding coefficient here if the common ancestor was not inbred. I hear 1/4, right? Is that what we see? So we have, let's just keep it though a little bit for later. How many, the inbreeding coefficient of I without the common ancestor being inbred? 1, 2, 3, right? So F of I equals, when we count the individuals in the inbreeding coefficient, that is, when there's 3 of them, it equals what? 1/8, right? For one loop, do we have another loop?
Yeah.
What do you think?
Plus 1/8.
Okay, so we have another loop, right? But that's not going to be influenced by the common ancestor. Yeah, so this is where it gets a little more complicated. So first we're going to figure out with this one, right? So it's 1/8. So that component of it, right, of this loop, of the inbred loop, right, is going to be 1/8 times 1 plus 1/8, right, plus this second loop of a non-inbred common ancestor. What does that equal? 1/2 to the third. That's going to be 1/8, right? All right. So that's going to be your total formula. So we take you through, and it gets a little bit more complicated as we go. I'll let you write that out, and then we'll do the math for it. Okay, so then let's go through and do it. F of I. F of I then equals 1/8 again times 9/8. Does everybody see that? 9/8. Does everybody see that? What is it? 1/8 times 9/8 is 9/64 plus 8/64. Does everybody see where I got that? I just expanded that 1/8 out. That equals how much? 17/64. If we were to compare that when the inbred, if there was no inbreeding, what was that answer before? For the full sibling? What's the inbreeding coefficient? If there's no,
The one we just did?
say it wasn't common ancestor, say full sibling matings, what's the inbreeding coefficient? Just let somebody look back. It's 1/4, right? Or if we expand that out, what would it be? 16/64. So by having one of the ancestors that was also inbred, we just increased it from 16/64 to 17/64. Corey, question?
Where's the third 1/8 come from?
This one right here?
Yeah.
This third 1/8 of the formula comes from calculating this loop right there.
Didn't we decide that wasn't?
You still have a common ancestor, right? And when we do the full sibling matings, we had to count for 2 loops. In these 2 loops, so we'll call this loop 1. Here is loop 2. With loop 2, we have to use a separate formula because, the expanded version of the formula, because the common ancestor in that loop was also inbred. And so that's why we had to add the components of this one. Okay?
Like in loop 1 though, is that whatever? Okay, cool.
So loop 1 is our traditional sense where there's a common ancestor, right? But that common ancestor, maybe that common ancestor moved into town from St. George, new arrival to Colorado City, so it's not so they're not inbred, right? And so he comes in, okay, that's great, but CA maybe is within that inbred community, and so then you're not influenced as much by it. Pat?
If the common ancestor was not there, then we would not have it.
Okay, so the question is, if the common ancestor was not where?
Like, connected to that, we would not have it.
Which common ancestors? There's these two common ancestors. Which one are we talking about? The male? Yeah. So if this common ancestor was not what?
Connected to the top circle.
Or another way of saying connected to the top circle was not what? Inbred. You wouldn't do that, right? That would just be like we did it 3 or 4 slides ago. Does that make sense? Okay. Now let's move on to some more complicated stuff. No, more practical stuff. So, for instance, I had mentioned that article in the New York Times that came out like 14 years ago when you all were in elementary school or whatever. And it actually was looking at the real effect of inbreeding. So we can do this theoretical calculations, right? We say, okay, this is the increase. But as medical professionals, right, you also just want to say, okay, what is the overall practical implication of that? So if we're looking at first cousin matings, we would say, okay, let's just, there may be more happening that we're actually calculating, et cetera, et cetera. We can see where we're getting more homozygous individuals, but let's do a big study and say, okay, in known first cousin matings, what is the increased incidence of, deleterious recessive allele conditions or different, physical manifestations of this inbreeding. And so then they did that in a big study, and it came out that, in fact, with first cousin matings, the total result of that was an increase in, disease or conditions of about 2 or 3% over outbreeding. So 2 or 3% increased incidence of these different things. And so is that a lot?
Yeah.
Maybe, maybe not. What is that comparable to? And this is I got this just simply from the article. That's comparable to the increase of incidence of trouble, of increased individuals, of increased offspring with these conditions, when you compare a female that gets pregnant between 20 and 30 compared to a female that gets pregnant after 40. So it is an increase, but is it as big a deal as we thought? That's why this article was kind of popular, because it doesn't seem to be as big a deal as not. And so then they had a discussion about the legal ramifications. In a lot of cases, it's illegal to have a child or to marry your first cousin because of this increase, right? But is it illegal for females to get pregnant after 40? No. So should it be? I'm not. I know it seems like I'm advocating here for first cousin marriages, but it doesn't seem to be as big a deal as we thought it was. Okay. Would that increase with half, with not first cousin, but say half siblings or uncle nieces or whatever? Yeah. And so that threshold, that first cousin threshold, and that kind of gives us a reason or some justification by why that first cousin mating has fluctuated with when it's been taboo with cultures and not, because it doesn't seem to be as big a deal as we think. Okay. All right. So now talking about practical effects. Can we calculate or quantify the practical effects of this? And this is what we want to do. So let's suppose we have a carrier for a rare deleterious allele, right? So we'll just throw out a number there. Let's say we have this deleterious allele that in the population has a frequency of 1 in 10,000. So, pretty rare. What is the probability? And there is a really, really good chance that all of us in here have a rare deleterious allele in our genomes that's hiding out as a heterozygous condition in our genomes. Not only one, but there's a good chance we have maybe a half dozen of them, maybe even a dozen of these rare deleterious alleles, right? And there's going to be varying degrees of deleteriousness to those alleles or whatever. But let's just pretend that, eventually we're all going to be grandparents, and we have, we end up having a daughter and a son. Could we calculate the probability with no inbreeding going on that one of those, we're just going to trace one of those alleles, that one of our grandchildren from either our son or our daughter is going to suffer from the homozygous condition, phenotypic condition of that? Could we calculate that? Everybody's answering yes. Let's see how would we do that. We haven't done any of these problems before, but we have enough in our sort of probability arsenal that we could calculate that. So let's try to calculate that. This is maybe going to be the last slide for today. Let's try to calculate that. Take a minute and try to calculate. Does everybody understand exactly what we're calculating? We're calculating, so let's say an individual is a carrier of this rare deleterious allele. This rare deleterious allele has a frequency in the population of 1 in 10,000. This individual has a son and a daughter. What is the probability that that individual will have a grandchild with that condition? No inbreeding going on. And then we're going to calculate it with inbreeding going on, and we're going to look at the difference in the probability. Those are the two. Okay, let's try to rebuild him. We're able to calculate that, so it's a really low chance. If in that first scenario they are outbred, it's a 1 in, what did we decide? 1 in 40,000 chance that the grandchild will have that condition. What is the probability if the son and the daughter decide for some reason that they're going to get together, mate, get married or not, and have the offspring? Now calculate that. How much does that probability increase? So now that's going to be your take-home assignment. Go through and figure out what's the probability if your son and daughter mate each other that you're going to have, that your offspring, your grandchild, is going to have that deleterious recessive allele. And decide how much it has increased in that scenario. All right, so see you on Tuesday. Okay, so now what we want to do, though, is we want to take it even up a level to being more practical. So that might have been kind of practical, that last one we did. But this one is, this is really what sort of scientists in the field, or maybe agriculturalists, or people that are doing breeding programs with cattle or horses or whatever, right? This is maybe more the kind of thing that they would do, or in this case PKU in the medical profession. So we want to see what the real effects of inbreeding are. Okay. What we see then, we have these numbers that we can calculate, these inbreeding coefficients, and they give us this sort of theoretical probability type thing, but we want to be able to translate that into some real effects. And we can do that if we measure the proportionality of that inbreeding coefficient and tie that to some real differences in incidences of certain conditions. So in this example, we're going to do PKU, phenylketonuria, among non-relatives. We can see the frequency of PKU in the population, right, when there's no inbreeding going on. And you can go out there and you can measure it and do whatever, right? And so you get that and you say, okay, the frequency of PKU when there are non-relative matings or no outbreeding going on is 1 in every 10,000. Okay, that's sort of a baseline level. If you were a researcher, you could then go in and look at a whole bunch of pedigrees and sort of data mine the information that's out there and say, I want to see what is the incidence of children born with PKU when they are the products of first cousin matings. And you would go in there, you'd research that, and all of a sudden you would find that, oh, the incidence of PKU has gone up, fairly significantly, 7-fold. So rather than having the condition in 1 in every 10,000 individuals, you now have the condition in 7 in every 10,000 individuals. That means the increase in frequency of when the inbreeding coefficient of first cousins is 1 in 16 is 6 out of 10,000 individuals. Now you might want to say, okay, maybe we have a lot of data, right, for first cousin matings, because that's sort of that borderline area, right? Both from social constraints and things like that. But let's say you wanted to then propose, okay, what would the expected frequency of half-sibling matings? If you were, say there wasn't a lot of data out there, but you were wondering, okay, what is that? What would we expect that to be for half-sibling matings? And then, okay, what would we expect that to be in full sibling matings? Because you have to find individuals that are carriers for PKU, and then also are full sibling matings, which is obviously going to be much more rare than first cousin matings. So could you do that? How would you do that? This is the next sort of problem. How would you do that? Should we try to do it? Let's try to do it try to see if you can come up with an answer for this first one in the next, let's say 30 seconds. Okay, let's see how we would do this. Does anybody recognize what that sort of little story problem had in it? It had how many data points? Three. Well, what were the 3 data points? It had 1 in 10,000, right? It had 7 in 10,000. It had an inbreeding coefficient of 1/16, right? With that first cousin mating. Did it have another inbreeding coefficient in there? What's the inbreeding coefficient when there is no inbreeding going on? 0, right? So really what we have, and you can see up here it says coefficient of, here let me move the timer so you can see it. Inbreeding coefficient over 16, and then frequency of something over 10,000. So in the first case, where they were outbred, what was the inbreeding coefficient? 0, right? What was the frequency? 1 in 10,000. But to make it a little bit more manageable, I'm just going to write down the numerator and not worry about the denominator, the 10,000. Okay, and so what would it be here? 1. What was the second one? 1 in 16. So you could write down 1 in 16, but I'm going to make it a little easier too to work with. I'm going to say we're going to do the inbreeding coefficient over 16 and just put the numerator again. So 1 over 16, I'll just put 1 here. What is the second one? Frequency over 10,000? 7, right? So what do we have here? We have pairs of different numbers that go together. We have essentially coordinates from algebra, right? We have coordinates that we could then draw a line from. So you could say here, these are our x coordinates. These are our y coordinates. Okay, so if we remember way back when to Math 1050, what was the equation for the line? Y, and this is just, all this equation for the line is doing is describing this relationship between the two variables, right? The x variable and the y variable. And so this is just a way of doing it. I've had students that do this on these problems and do the line and are great. I've had students who just kind of look at it and then just kind of write the next ones down. So some people can just kind of look at the increase from here to here, which is what in your equation down here?
Slope.
It's your slope, right? It's your m. So this is y equals slope times x plus b. What is b? Y-intercept. Okay, y-intercept where x is 0, you have b. So based on our coordinates here, where x is 0, what is y?
1.
What is the intercept? 1. So our equation then becomes, I'll just write the equation over here, y is equal to mx plus 1, right? So now what is m equal to? We have the equation of the slope over here. M is equal to y sub 1 minus y sub 2. Where's y sub 1? Here, y sub 1 is 1, y sub 2 is 7, right? Over x of 1 minus x of 2, x of 1 is 0, x of 2 is 1. So you get m equal to 1 minus there's our relationship right here. We have it. So X is the inbreeding coefficient over 16, right? And so what was the first one? It was half-sibling matings, right? What is then the coefficient, inbreeding coefficient for half-sibling matings? 1/8. Well, let's keep it consistent over here. Let's just say it's 2/16, right? And so here our X would be 2. Does everybody see that? Instead of 1/8, we're talking about 2/16. This is just me. This is just the way I do it because I like to have things like more simple. So here's 2. If we plug in 2 to here, 2x in this equation, y equals 6 times x, or 6 times 2 plus 1 is equal to what? 13. Okay, there you have it. There's your answer. And you could actually, you know, if you had paper, you could actually chart it out 1, and then here was, I don't know, I'm just going to 7, and then here was 13, and you could actually draw a line and be able to, you know, come down here on X where it's, let's say, you're given a number here, and then bring it up here, and you'd have a pretty good estimate of what that effect would be. Okay, what was the next one? Full sibling, right? What's the inbreeding coefficient of full sibling? 4/16. Plug 4 into there. What does that equal? 4 times 6 is 44. What did I hear back there? 4 times 6 is what? 24. Plus 1 is what? 25. Okay, good. So it just becomes a pretty easy, I mean, yeah, maybe it's been a little while since you've done your algebra, but it essentially just becomes a pretty easy little algebra. A pretty easy algebra problem. Some people can just go here and say, okay, moving from 0 to 1, there's an increase in 6. Going from 1 to 2, an increase in 6 more. 2 to, well, you can even plug in 3 here. Increase of 6 more, be 19, right? 4, increase of 6 more, 25. Okay, so some people, you can just draw that out there. Some people like to draw a line. Not too many people like to draw a line, actually, but you can. And some people like to do the equation. Most people will do the equation. Well, that's split between just doing it this way and doing the equation. Okay, any questions on that one? So that's what are we doing here? We're just describing this relationship between the inbreeding coefficient that we kind of have the theoretical background for. We know where that's coming from, right? But it's just kind of assigning a real world practical interpretation of what that inbreeding coefficient is doing. Here are some charts of, I gotta pull this out. Here are some charts of real world data with plant height and crop yield and different inbreeding coefficients. So down here we have an inbreeding coefficient all the way from 0 to 1, 0 to 1. We know that in let's say if we were looking at Mendelian pea plants, right, the inbreeding coefficient, the highest inbreeding coefficient in one generation that you can actually have is a 1/2. So if, like with Mendel's pea plants, they're breeding, then, or it's there's self-fertilization going on, it's 1/2. And so then how do you think you get all the way up here to close to 1? How could you get an inbreeding coefficient there? How do we increase an inbreeding coefficient? Decrease what, Carl? Yeah, closely related, you know, maybe with diploid organisms, your closest relative is who? Yourself, right? So you're breeding with yourself if you're the pea plant. You have an inbreeding coefficient of 1/2. Where could we increase that? How are they getting these here's 1/2, right? This is the effect of 1/2 on plant height. It's 165 centimeters. Over on this side, crop yield is only about 30 kilograms per 100 meters square of field. How would you do that? We know how to do it. We have the knowledge. Deep in your brain, you have the knowledge. When have we seen increases in inbreeding coefficient? When the individual, the common ancestor is also what?
Inbred.
Yeah, when the common ancestor is inbred. Okay, so if you have offspring, say you're a pea plant, you have offspring, you're the father/mother of that offspring. And if you were inbred, or say you decide, okay, I'm going to inbreed, I have a bunch of little peas. Those little peas decide, okay, we're going to self-fertilize too. How would we calculate the increase in the inbreeding coefficient? Do we know the formula? Inbreeding coefficient equals F times 1, good, plus, the inbreeding coefficient of the common ancestor. Okay, so there's that equation. So if you're inbred, let's say generation 1, let's have this first generation, selfing, right? That inbreeding coefficient is 1/2 because you're selfing, right? Anytime you're selfing, you have an inbreeding coefficient of 1/2. Now in this next generation, that individual is going to be inbred, right? So the inbreeding coefficient without a common ancestor that's inbred is 1/2 plus 1 plus what was the inbreeding coefficient of a common ancestor, which is themselves. It's kind of weird, I know. It's 1/2, right? Add those together, 1/2 times what? 3/2, right? 3/2 equals 3/4. Okay, now we've just increased our inbreeding coefficient above the 1/2. What would happen in the next generation? What would the inbreeding coefficient for the next generation be? 1/2, right? Times 1 plus what? 3/4. Let's do this one. Times 3/4. What does that end up being? 1/2, right? Times, add those together, what's that? 7, how much? 7/4. Times those together. Then, oh, you mean the total, yeah. The total is 7/8. Right now we're going to increase from here to here. And as you keep going up more and more and more, you begin to approach 1. So that's how they got these inbreeding coefficients above 1/2. Okay, so remember that. Inbreeding coefficient of a self-fertilizing individual is 1/2. How to get that above that? And here are the actual effects. And so you see this dramatic decrease in plant height from 190 centimeters all the way down to 150. Pretty significant decrease with that increased inbreeding. And then think back to those pictures of the corn. Just think of maybe, I don't know if this is exactly, yeah, I think it's corn. Maybe not. Think back to those pictures of those inbred lines, and you can see where the inbreeding has that actual real world sort of effect. Okay, let's talk about population genetics for a second. So if inbreeding is this march towards homozygosity, what does it do to a population? We all know, or we've all heard or read about what inbreeding, lots of inbreeding does to populations that are sometimes threatened, or populations that are in of animals that are in zoos. For instance, one way that, another way that inbreeding occurs in natural populations is if natural populations go through what we call a bottleneck. How many of you had ecology, or how many remember talking about bottlenecks in ecology? What happens to the population? You get an increase in homozygosity. After the bottleneck, because the population is really small, you then are forced to inbreed, right? So if you get a population down to, 1,000 individuals or 100 individuals, in a few generations, it's very likely that you're going to be inbreeding, maybe even in the first generation. So you have this bottleneck plus inbreeding that decreases the genetic diversity, right? That's why they become more susceptible to extinction, is because you have decreased the genetic diversity. That's what inbreeding does also. So bottleneck plus inbreeding has done that too, and the effects of that have been, for instance, with the classic example of a massive bottleneck was with cheetah populations, Asian cheetah populations. We used to have cheetah populations here in the Americas a long time ago, but they went extinct in the Pleistocene about 12,000 years ago. But we still, so this is a total side thing, but we still have evidence of those those cheetahs that were once here. Does anybody know what living evidence. What's the living evidence of the cheetahs that were once roaming across Wyoming? Cougar? No. You mean cougars evolved from those cheetahs? No. Or what?
The ability of certain prey to run real
Yeah, certain prey to run really fast. What if you've ever driven across I-80? What do you always see? Has anybody done that drive like to Laramie? What do you see? Yeah, you see tons and tons of antelopes. And antelopes are so much faster than any potential predator. Biologists wondered, why have they evolved these really fast? And
They could live the behavior where they, I can't remember what it's called, but they jump straight up. The stotting, which is the behavior that's an animal in Africa do as well when there's a predator.
yeah, so you have all of these leftover remnant things that served the antelope population well when we had a cheetah over here in North America. Different species, but a Pleistocene cheetah over here, they're super fast speed. There was this evolutionary arms race between the cheetah and the antelope. Cheetah gets faster than the antelope, cheetah, etc. Same thing that happened in Africa happened here, but then all of a sudden the cheetahs all died. And so now you have these antelope that are super, super fast, basically way too fast than they should be, right? So anyway, okay, back to cheetahs. So the interesting thing about cheetahs, though, is the African cheetahs also went through a massive bottleneck, a massive reduction in their genetic diversity, and since then, and this bottleneck happened about the same time that the ones here went extinct, about 10 to 12,000 years ago. So cheetahs have so little genetic diversity that you can actually do skin transplants from one cheetah to another cheetah, and there's no rejection. They don't have the rejection of the skin because they're so uniform in their genetic makeup. So that's what's happened. Population, in this case, either founder effect or bottlenecking in ecological terms, followed by inbreeding. You see that what happens in a regular population, and this is just a mathematical sort of explanation of it. Here we have generation. The first generation, we have the allele frequency. So we're talking population genetic growth, talking about allele frequencies. The allele frequencies of the big A, little a alleles is 0.5 and 0.5. Does everybody see that? Do you remember this from 1110? How do we figure out the allele frequency in that generation? Each of these individuals have how many big A1 alleles? 2, right? So there's 500 alleles here. How many alleles are there here? How many A1 alleles? 500. How many A2 alleles? 500. How many A2 alleles are here? A2 alleles here? 500. So what's the total number of alleles? 1,000 in that population. Well, no. How many total alleles? 2,000. How many big A alleles in that population? 1,000. How many A1 alleles? 1,000. How many A2 alleles? 1,000. Therefore, the frequency is 0.5 and 0.5. Right? That's in our regular population. But let's go to extreme inbreeding. What happens in extreme inbreeding? You mate with yourself, right? Let's say these are pea plants. If all of these individuals mate with themselves, what are the offspring going to be? Yeah, A1, A1, right? All of these, A1, A1. But if these all mate with themselves, the heterozygous individuals all mate with themselves, what's the ratios going to be? Yeah, good. So 1 to 2 to 1. 1/4 of them will be A1, A1. 1/4 of them will be A2, A2, and then half of those will be the same heterozygous. So what's happening? Those A1s on that selfing get dumped into here. The A2s get dumped into here, and essentially the number of heterozygous individuals half. Is the allele frequency different in this next population?
Yes.
No. You can do the calculation. It's not. It stays the same here. It stays the same here. But essentially what happens is each time you go through that generation of self-fertilization, that massive inbreeding, you lose heterozygous individuals. What does that do to your genetic diversity? That causes it to go down and go down and go down. What problems does this have in the population? You're reducing your genetic diversity. You have more homozygous individuals. What does that do to your population? Not just reducing your genetic diversity, but what else could it do?
It increases
Yeah, because of the genetic diversity. And what else? What other practical effects, like we were just looking at just barely? If more and more of your population is homozygous for these alleles, you have a much higher probability of having diseases, because you have more and more individuals that are homozygous for these deleterious recessive alleles, right? That's going to affect things like plant height and all of that other stuff. Okay, so a second aspect of inbreeding for population genetics is that we that when we have outbred populations and we have recessive deleterious alleles, we can hide out as recessive deleterious alleles, because the more rare in the heterozygous state, because the more rare the allele, the less likely those alleles have, less likely chance those alleles have in forming a homozygous individual. Okay, and this is just an exercise that we can do that will show us why that is. Here is an allele, a frequency of 20% here, 2 in 100 here, and 2 in 1,000 here. So this is a not so rare allele, right? It's not the dominant allele, or it's not the most frequent allele, not dominant, the most frequent allele, 20%, but it's not that rare. So if we were to calculate this and we were to look at Hardy-Weinberg equilibriums, does everybody remember? We kind of did Hardy-Weinberg equilibriums earlier this semester when we were doing the probability, right? We did the equation. Does anybody remember what the equation was? When we were doing the flipping the coin and the alleles, if we were to flip heads, what would that equal? P squared, right? 1/2 times 1/2. What was the frequency of the tails? Were the other homozygous? Q squared, right? What was it? So 1/4, right? Getting heads, 1/4 getting tails. What was the probability of getting a heads or a tails? P cubed, but there were two combinations, 2p cubed. Remember that? That's the Hardy-Weinberg equilibrium. There's a whole bunch of assumptions that go into the Hardy-Weinberg equilibrium, that they're randomly mating, that the population size is large, etc. But if there's no selection going on, there's no migration, there's no mutation. These are all the assumptions of the Hardy-Weinberg model. Under the Hardy-Weinberg model, though, if we're given an allele frequency, we can calculate what are these in the Hardy-Weinberg model? Genotypic frequencies. Okay, what's that? Yeah. So this one is this is the homozygous dominant, right? This is the heterozygous individual. This is the homozygous recessive. So if little p, or we'll just call it f of a, we'll just say, okay, f of a is equal to p, which is equal to what? What's the frequency of little a? 0.2. So now we have our p value. Sorry, we're doing q. That's geometry. Therefore, the frequency of big A is equal to p, which is equal to what? 1 minus 0.2.
1/8.
Yeah, 0.8. Okay, there's our p. And now with our p and our q, we can just plug and chug into that equation, and we can figure out, okay, what proportion of the population is going to be in each of these. And so what we see, and we won't take time to go through each of these, but maybe you can do it on your own now that you kind of know how to do this, we'll see that when we go through and calculate each of these, that this q squared, right, is going to be smaller and smaller by order of magnitude as we go from this situation to population 2, where f of a is equal to 0.02, to population 3, where f of a is equal to 0.002. Pat? Yeah, so if you have two alleles in a population and allele A is 20%, you know allele B has to be what? 80%. So that's where you get that. So you can go through, plug those into the Hardy-Weinberg equilibrium, and you will see that as you do that, it becomes more and more. I've done it already. Sorry, should I just write them up here for you? I'll write up 2 of them for you. Okay, so I already went through and did all the calculations. So population 1, population 2, population 3, little a, big A, little a, big A, big A. In the first population, 64% are big A, 32% are heterozygous, and 4% are little a, okay? So you have about 4% of those homozygous recessive. In the second population, 0.0004 are little a, 0.039 or 4% are heterozygous, and 0.96 are big A. Okay, so you see a trend going on here. Is it very likely that you're going to have little a individuals in the population? No, not so much, right? In this population, if this population then was randomly mating, would you get little a individuals out of there? Yes, if the little a got with a big A, right? Or little a got with another little a. Or when you had the heterozygous individuals mating, you have a probability there to get the little a out of that mating. Okay, so in the third one, 0.0000000, I think I have it right. 4, 8, or no, 7 zeros here. So very, you're hardly ever, essentially never going to see homozygous individuals. So you're not going to see that condition. So each of us has some, recessive deleterious allele in our genome that we don't see because it's such a low probability that you have a little a, right? Here, heterozygous individuals, 0.004 essentially there, and then 0.9996 there. Okay? So as the allele gets more and more rare because of population genetics, we see that we end up having individuals that are we think it's pretty much everybody is almost uniformly. Remember we said the rare alleles in the genomes paper, is it anything over 1%, is it? Do we even count the allele? You guys remember back to the first lab? I think it's 1 or 2%, right? Here that wouldn't even be counted. So we wouldn't even know that allele is in the population. But if you begin to get inbreeding going on, you're increasing this by orders of magnitude. You're increasing the probability of getting a little a by orders of magnitude. Okay? And so that's the difference. Okay, so the last thing that we're going to talk about is measuring genetic relationships. Luckily, you all already know how to do this because it's just a step 1. You just add 1 step to calculating the inbreeding coefficient. But it's a very, very important thing to know. When we're calculating our genetic relationships, we know there are certain people in our lives that we're more closely related than we are to other people, right? We know that we're more closely related to our siblings than we are to our second cousin, right? So let's say you're Leonardo DiCaprio, you're on the Titanic. There's a whole bunch of people coming into the lifeboat, right, before it goes under. There's that big iceberg right there. So this is a very real world example. You are confronted with having to bring in your mother or your sister. There's only one space, and you can't give up your own space because you're the only one that knows how to drive it, or you don't drive it, whatever. You steer it, right? Do you let your mother on, or do you let your sister on? Raise your hand, mother. Yeah, we have some issues in here. Raise your hand, sister. Everybody ever let their sister in? Okay, that one. Why is it? Why, Maya? Why your sister?
Well, I mean, your mom is probably done reproducing, but your
Oh,
sister,
she's thinking about it in evolutionary terms.
but
Good.
your sister can keep passing on your genes.
Okay, good. Okay, how about your first cousin or your niece or nephew?
Have your niece.
This one maybe is a little bit more, let's just say not even Titanic. Christmas or holiday presents. Who do you give better presents to? Your first cousin or your niece? Yeah, most people give to their nieces and nephews, right? Okay, so this class can help you with these decisions. And how do we do it? We do it by calculating our genetic relationships. So the calculation of genetic relationship is a term that we call the coefficient of relationship. Essentially, it's saying, okay, how many of my genes, and this is important. Maya started talking about this in evolutionary terms, and this is where it's important when you're trying to figure out behavior. A lot of time we go out into nature and we see what we think are these altruistic behaviors, when in reality, so for instance, the classic example is the beehive or the ant pile, where you have a whole bunch of non-reproducing individuals, right? Why do they do that? Why do they sacrifice their own reproduction to help out the queen ant or the queen bee reproduce a whole bunch? And it has to do with this coefficient of relationship. So even if you're not passing down your own genes, you can vicariously be passing down your genes through shared genes with other individuals. And so this is how we do the coefficient of relationship. You essentially just hypothesize, not a very happy thought, but you hypothesize inbreeding between you and whatever that individual is, your niece, nephew, cousin, whatever it is, right? And pretend that there's inbreeding going on there, and you calculate the inbreeding coefficient. You then times it by 2, and that tells you, on average, how many of your genes you are sharing with that individual. So, for instance, back to first cousins. So, with your first cousin, if you were to mate with your first cousin, right? What is the inbreeding coefficient between matings of first cousins? Remember, back 1/16 times that by 2, 1 in 8. So that's essentially telling you that an eighth of your genes on average are shared with your first cousin. Okay? So here, full siblings. Full siblings, let's do this now when you're on the Titanic, deciding between your brother and your mother, or your sister and your father. We're not taking into account mitochondrial genes either, because those are passed down differently. Because then you would favor your mom, because she has that little added part of it, right? So full siblings, your inbreeding coefficient is 1/4, times it by 2, that equals 1/2. Essentially, and we kind of know this intuitively, how many of your genes on average do you share with your siblings? Half. How many genes do you share on average with your parents? Think about the chromosomes, you can think about meiosis. You're getting half of your genes from your mother, half of your genes from your father. And so you're just as related to your sibling as you are to your parents. So maybe you'll want to do what Maya did and say, okay, who has the better chance of increasing, your reproductive potential? Okay, so can we calculate our evolutionary fitness? What do you think? And this is just a diagram kind of showing you those pathways. So how can we calculate our evolutionary fitness? Who's in the next generation? Obviously your kids, right? So if you have 2 kids, right, how much of your genome is going to be represented in the next generation through those 2 kids on average? Half through one, half through the other. Therefore, you're represented, right? Pretty much on average. If you have a 3rd kid, anybody have 2 kids thinking about a 3rd kid? If you have 2 kids thinking about a 3rd kid, maybe this will sway your decision. You have a half more, what does that mean? 1.5. Therefore, are you gaining? Are you winning evolutionarily? Or are you losing? You're winning, right? So you have a 3rd kid, you're 1.5. You have a 4th kid, you're 2. Yeah, so keep going, right? Because you're getting more and more evolutionarily fit. However, some people maybe don't have any kids. Does that mean that they don't have much? They're not very evolutionarily fit? Not necessarily, because you share if you have siblings, you're sharing half of your genes with your siblings, and then they're having nieces and nephews. On average, how much of your genome do you share with a niece or a nephew? 1/4, right? So I had a friend in graduate school. He was from Missouri, and he was one of 11 kids, youngest of 11. He married a gal from Malad, Idaho, or someplace up in Idaho. She had 12 siblings. They couldn't have any kids, but they had 103 nieces and nephews, or something outrageous like that. So, how are they doing evolutionarily? Not too bad, right? So, how do you calculate that? Could you calculate your own evolutionary fitness right now? Yeah, and you're going to. And then when you come back, we'll see. Oh, I guess that's after the exam, though. So, an assignment for next week, we're going to see who's going to be dubbed the most evolutionary fit person in this class.
Wait, does it go by who has the most siblings and nieces?
got to calculate it.
What if you don't have any nieces or nephews?
And or kids.
No.
You should just talk about your reproductive potential because you don't have any yet. You haven't actualized any of it yet, but you could. Oh yeah, your brother or sister's not married. Do you have a sister?
Yeah, and neither of them have kids or anything.
So you could even go even further out, right? How about first cousins? If your first cousins have some kids, you'll be passing down a little bit down there. So I want everybody to try to generate. No, I know what we'll do. We'll do it now, and then we'll talk about it in lab, and we'll see in lab who's the evolutionary most fit. Okay, so before we go, I think. All right, so what I want you to do is I want you to calculate your evolutionary fitness by calculating the coefficient of relationship or relatedness with your children and your nieces and nephews, and then adding it all up, and then we'll see who has the highest evolutionary fitness.